Good morning, everybody. My name's Edward Abrahams. I'm the president of the Personalized Medicine Coalition for another couple of months. And it is my pleasure to welcome you to our 18th annual meeting at Harvard Medical School. Our first meeting was here in 2005. And I think that this is a pivotal moment for personalized medicine, as we've written in the introduction to the conference in the program. And it is a pivotal moment in part because the science and technology that underpins personalized medicine, as in 2005, continues today to far outpace our ability to integrate it into healthcare. Which is the reason PMC was created, was to address that particular problem. We are, as I think most people here know, dedicated to addressing the issues between the science and the patient. And as such, in my opinion, we are the only organization so focused. And we have a lot of work to do, and part of what this conference is about is to think about those issues, which tend to be government related, but not only. They also involve education and the clinical adoption by providers and particularly payers. So at this conference, you won't hear, I hope, a lot of company presentations. You can get that elsewhere. But here we're going to focus, as PMC does, on, again, that space between the science and the patient. Our goal is to solve the problems to bring personalized medicine to patients. And I would also humbly suggest that AI is not going to solve that problem, that AI might even stimulate more opportunity, but unless we address the issues that we're going to be talking about today, we won't be as successful as we might otherwise be. And I think also PMC is well situated to think about this ecosphere because, as represented in this room right now, we are a diverse coalition of stakeholders. So that is what I think of as should frame this conference. And when we go through the different panels and presentations, I hope you'll think of it in that context. I especially want to turn my attention to thanking the people and institutions that make this conference possible. First is Boston Children's, which has partnered with PMC to organize this annual meeting, and we're particularly thankful for them to making this facility available to us. The sponsors of this conference not only underwrite the conference and the reception you'll go to tonight and the nice food, et cetera, but they also underwrite the work of PMC, and I'm deeply thankful to them, especially our lead sponsors, Amgen, LabCorp, and Pharma. I certainly want to thank Chris Wells, who is the conference director, along with the multiple other things he does at the Personalized Medicine Coalition. And I would be especially remiss if I didn't thank Jen Kazenza, who does all the logistics for this conference, but equally important, has been with us I believe since the beginning, going back to when we were working with a company called Feinstein Keene. So Jen has been instrumental, as has Nicole Jacobson, who also helps us put this conference on, which I, for one, don't take for granted that we have such great people working for us. I also wanted to introduce, and I'm hoping she's here, the new president of PMC, Amy Nicole Nair, who comes to us It comes to us from Novartis and cell and gene therapy, which has a long background in advocacy. And I'm certain she's going to be taking PMC to new and higher levels, especially with everyone's here's help. And I've told her she can count on that. It's now my pleasure to introduce Clifton Leaf, who's a former editor-in-chief of Fortune magazine, but for my purposes, helps us understand and why we asked him to do the first two interviews of the conference. He helps us, as I say, understand the ecosphere of healthcare today and personalized medicine in particular. So with that, I'd like to ask Cliff to come to the stage and introduce our first speaker. Thank you.
Is that better? That's much better. See, I'm a pro. I know how to turn on a button. So David's full bio isn't here, but I just love to get him up here and we can and talk about this amazing revolution that we're witnessing. Pro tip: turn on the button. Yeah. David, I alluded to the fact that you were at the forefront of this, and in our conversations, in our earlier conversations, I discovered that you were in the lab, working in the lab with your two mentors, Denny Slayman and John Glaspie. The Slayman lab at UCLA was the lab that discovered the human HER2 and its relationship to cancer, which was really, if not the beginning of the personalized medicine adventure, it's certainly an early milestone, an important one. Tell me a little bit about that perspective of seeing this genetic connection where you could pinpoint a subset of patients who were at severe risk of this disease.
Yeah, in fact, the fact that I'm sitting here is probably due to a coincidence. My first rotation as an intern in the internal medicine program at UCLA was on the oncology wards, and Dennis Slayman was the attending physician that month. Just a couple months earlier, he had published one of the seminal papers identifying HER2 as a human oncogene and a driver of a subset of breast cancer. At the end of the month, he said, you seem like a reasonably intelligent guy. Why don't you come by the lab and talk to us? And I ended up spending a decade in the lab when the work was being done to, fully flesh out the role of HER2, and then the work that led to ultimately Herceptin preclinical work and clinical work. So in a lot of ways, it was the birth of translational medicine and the notion of going directly from a target to a therapeutic.
Yeah, I mean, it's extraordinary. And we've talked about with this lens of history that you have, we're at a new moment. We're in an era. You call it the sort of hinge moment. And I just want to make sure that anyone from the Patent and Trademark Office knows that David actually coined that term years ago. So we want to get credit where credit is due. Yeah. Tell me what you mean by the hinge moment.
Yeah, I'm now our chief technology officer. I've been at Amgen for 19 years. My role prior to this was as the head of R&D for about 6 years. And half a dozen years ago or so, I started talking to our board of directors and CEO staff about what I called the hinge moment, this notion that we're seeing a revolution in technology, and technology or tech and biotech were coming together. And that was going to transform drug discovery, drug development, manufacturing, and commercialization. So the entire spectrum. And
Hasn't it always been though this way? I mean, you go back to Van Leeuwenhoek and his crazy microscopes and, grinding 500 different lenses. I mean, we've often thought about biotech, the sort of biology and technology together. What's so unusual about this sort of confluence of new technologies?
Yeah. So if you take the historical perspective, in my academic career, I was an amateur medical historian and wrote a few papers in this area. Look, there's always a constant back and forth between technology and science. Technological advances lead to new scientific insights. Those scientific insights in turn drive the development of technology. Obviously, the invention of the microscope, then the perfection of the microscope in the mid 19th century gave rise to the cellular theory of medicine. So we finally threw off, 2 centuries or two millennia of Hippocratic medicine, and the notion of the four humors. So it's our belief that now we are in one of those once every generation or two moments where changes in technology are really going to propel the field forward. I think that's now undeniable. There are those who are betting against it. I wish them all the best luck.
And to drill down into this, AI, machine learning, CRISPR technology, gene editing, with single cell analysis. What are some of the ways in which these particular technologies are going to change the way we think or think about science and pursue research?
So if we start at a high altitude, what is really fueling this hinge moment? What is the crux of it? What you're just implying, data at scale. We're now generating staggering amounts of data. Last year, the world generated about 120 zettabytes of data. A zettabyte is 1 followed by 21 zeros. Why do we care about that? About 30% is actually in healthcare. So it's not all TikTok videos. And so now you have that massive amount of data, and now analytical techniques with the rise of generative AI in particular that allow us to go at it. Are these tools? Of course they're tools, but they're some of the most powerful tools that we've ever seen.
And one of the new breakthrough areas has to do with the sort of language of proteins intermingling with each other and binding. Talk a little bit about that and what Amgen specifically is doing there.
Yeah, so obviously, we're keenly interested. We're 44 years old. We're keenly interested in molecular engineering and the generation of protein therapeutics. And when DeepMind published AlphaFold, that was a before and after moment. We literally had downloaded the code. We were working with that material within hours of publication. Fast forward to a few weeks ago, we released what's called a protein language model into the public domain that we developed with an institute, an AI institute in Montreal that can be of foundational use in molecular engineering. So one of our core challenges is, of course, we find a target that's of interest in a disease. How do we develop a therapeutic? And that has been largely empiric, meaning if you want to generate an antibody, we can very rapidly generate panels of several hundred antibodies. And then we have a rigorous screening process that those go through. We generate data, we then re-engineer, and you get these iterative cycles. What we're now doing is in silico design of portions of that process to greatly cut down the number of cycles of empiric experimentation that you need to do in the laboratory. One of our existential challenges is what we call cycle time. It's still 10 to 14 years from an idea in the laboratory to a drug, actually, if it's one of the 8% that's lucky enough to make it to marketing authorization. So you can take literally a year or two out of the preclinical development time through with the assistance of in silico design. We're just getting started. I'd sort of respectfully disagree with Dr. Abrams' remarks initially in terms of this being a game changer. I don't think if you pick the right problems, there's no question it's a game changer. Does it change some of the structural problems that we have in the system in getting, drugs to the right patients? No, it might actually be able to help there, but picking the use case is critical.
Okay, I just want you are the one that opened this discussion. So what are some of those structural problems? Yeah.
Well, when I became head of R&D at Amgen, we had sort of three strategic imperatives. Two of them are existential challenges for any R&D organization. Reduce the cycle time, which I just talked about. Right. Increase the success rate. If you can go from that 8 or 10%, I used to tell my son, if I failed 80% of the time, I would be the best ever by a mile.
Yeah. Yeah, absolutely.
So increase that success rate. But then I introduced a third pillar, which was enable access and use, meaning starting to think right from the laboratory, how are we going to most efficiently get that drug into the hands of the patient who needs it? Because the most elegant drug in the world doesn't do any good if it doesn't get to that patient. And so it's really thinking about that entire ecosystem. It can be something as simple as, can you get it into an auto injector that can be delivered by the patient at home once a month? That eases access and use. Can you demonstrate, overwhelming efficacy in a particular subset of patients, et cetera, et cetera.
And you talked a little bit about the sort of cultural barriers too, about the fact that, some patients who may be eligible for a targeted treatment aren't getting it for one reason or another. Either they're in the community setting and they're not getting, they're not getting their biomarkers tested. Their biospecimen doesn't have enough cells to get a good signal. There are a whole bunch of issues that are happening. But talk a little bit about what you see as some of the solutions for that.
Sure. I mean, if we think about what needs to happen, for a patient to get the right personalized therapy, there has to be the correct diagnosis. That's the foundation. Then there has to be the appropriate molecular testing of that specimen. If it's let's take cancer as the simplest in many ways use case. So the right molecular testing, but then that testing has to be put in front of the physician in a way that is digestible and understandable for that physician. And then there has to be translation of that information into the appropriate therapeutic plan. So we think about each step along the way and how you can help. Even with today's targeted therapies in lung cancer, where there's, of course, overwhelming evidence from clinical trials and practice of the efficacy of these drugs, in some settings, you still only 85% of patients are getting the right testing. So getting the test is the first step.
Mm-hmm. When the first draft of the human genome was published in 2000, shortly after that, everyone, and I mean everyone certainly in this sphere that we're talking to, thought, oh well, we're halfway home now. We'll understand the genetic basis for disease, not just in cancer, but through heart disease and cystic fibrosis and a whole bunch of other areas, rare diseases, and all we have to do is find that gene, and we'll be able to address that through a targeted medicine. We're now at a point where we've opened up the curtain to the proteome and the transcriptome and the epigenome. How many more of these omes will we have to discover before we sort of say, okay, that's the secret?
I would. So my son just passed the California bar exam, and so I now can seek legal advice internally. But so speaking like a lawyer, I would. I guess I would disagree with the premise of the question that there's the answer. There's not the answer. The answer is customized to whatever clinical setting we're talking about, whatever disease. Let me go back up to another altitude. I said, we're starting and we're embarking on an era where we have massive amounts of data, and we can now begin to mine that for disease insights. We acquired a company called Decode Genetics, now Amgen Research Reykjavik, in Iceland a dozen years ago. Founded to really collect genetic information. Now we have multi-omic data on up to 3 million individuals. It's roughly 300 petabytes of data. So genomic data, proteomic data on a subset, transcriptomic data, coupled with dense phenotypic data, everything from your hair color to your CT scan results. We just finished a few months ago the installation of an NVIDIA SuperPod, so an NVIDIA supercomputer, and we're now building foundation models to actually probe that data set. Why? Why is that relevant to this audience? Well, we want to ask questions about, for example, autoimmune diseases. A disease like lupus, my belief, and I think this is almost undoubtedly true, is that just like breast cancer or lymphoma can be categorized into relatively discrete subsets of diseases based on mutational profile, lupus for sure is multiple diseases under this crude clinical name that we give lupus, where we still use an 11-point check list that's decades old now and is unbelievably simple to diagnose the disease. I'd like to understand, what are those subsets? Why do some patients get skin-only disease? Why do some have terrible kidney disease? And that hopefully then leads us to understanding pathways and generating therapeutic hypotheses. That's a true personalized medicine, but we don't understand the pathogenesis of that disease. That's where the new technology can actually help us. And in fact, without machine learning and artificial intelligence, we have no hope of making sense of that data set.
So that Decode Genetics, the Icelandic biobank, is there a subset of those that aren't blonde and blue-eyed? And secondly, what did we learn from the genome?
Well, we have 3 million individuals, and there are only 350,000 Icelanders. So,
so you've moved beyond the Icelandic population.
And we're trying to constantly diversify the populations that are in that database. Which is very important, but there's something that's also important that's almost never said, which is humans are actually much more alike than they are different. They're much more alike than they are different. In fact, that's why it's so hard to find those differences and the ones that are important on a disease level, because they're often subtle. So you can extract huge amounts of information that are relevant to populations around the world, even from relatively homogeneous populations. That's not to say that diversifying these databases is not important. It's of critical importance.
So when you look at the last election we just had, you can say that we're more alike than we are different.
I think political commentary in this form is beyond my remit.
So in terms of, what do we learn from the fact that we're more alike than we're different? Like what can we learn from that, in terms of the pathology of disease or how to treat people or the risk of that some people are, have side effects that others don't? Isn't there the idea of the sort of pharmacogenomics, the individuality of each person in terms of reaction?
Yeah, sure. Exactly. And trying to find those differences is why you need data sets that are this large. But we have examples, for example, of genes that we found in a Japanese population that pointed us towards a disease insight. We then, of course, replicate it in a bigger database, and lo and behold, those variants are present in very low frequencies in Western European populations, but when they are, they segregate with the same kind of disease risk. So, so at a fundamental level, that human biology is the same.
Yeah. So in terms of what Amgen is sort of focusing on, this personalized medicine revolution, talk, if you will, about some of the programs that you have in-house that really speak to the specificity of a subset of patients that can be really treated with something targeted.
Let me talk about one of our cardiovascular programs,
Yeah.
which I think brings together a number of things that we were talking about. This is a program that targets LP little a, lipoprotein little a. LP little a is a lipid particle like LDL cholesterol, the bad cholesterol. Everyone knows about that. LP little a is probably responsible for about 30% or so of cases of atherosclerotic heart disease. It's probably the single biggest risk factor beyond LDL
It's
cholesterol.
stickier than LDL,
It's
right?
sticky.
Yeah.
Exactly. I was just going to say it's highly atherogenic, meaning causing coronary artery disease.
That was the word I was looking for, but
Yeah.
when I said sticky, yeah.
I was just
Yeah,
helping you out.
yeah.
And it is a genetically determined level.
Right.
About 15 or 20% of the population, so 15 or 20% of us will have elevated LP little a levels. Not only is it genetically determined, it's largely genetically fixed, meaning it's not modifiable. Diet and exercise have essentially no effect on LP little a, so it's different than LDL cholesterol in that regard. Currently available drugs have modest to no effects on LP little a levels. So we developed a small interfering RNA that essentially shuts off the gene responsible for LP little a production in the liver, where the vast majority of your LP little a is made. In phase 2 studies, it showed a 95% or greater reduction in LP little a levels, and it's now in a very large phase 3 outcomes trial. So that's a very, very specific hypothesis. Now, what's interesting is that as part of our work in cardiovascular medicine, we also began developing on the Decode database proteomic risk scores. Some of you may be familiar with polygenic risk scores. So collection of genes, it may be a handful, it may be up to 50 or 100 that collectively give you a risk score for a specific disease. It's been used most often in heart disease, atherosclerotic heart disease. What we were able to do with the proteomic data that we have, so this is a snapshot of proteins in your blood, their expression levels at any given time, several thousand of them, show that a subset of about 100 would generate a risk score that would increase your ability to predict risk of a cardiovascular event about 2.5-fold, which in that field is an enormous increment. So we're now actually embedding that risk score, we're validating it in trials that we have going now. Why is your proteome potentially more interesting than your genome? Your genome is essentially fixed. You hope it's fixed. Your proteome varies. If you had breakfast this morning, proteins in your blood actually changed in response to that. It can give you, in other words, a kinetic snapshot, and we have inklings that proteome changes over time, and certain changes are harbingers of bad things happening in a disease course. So now the goal is to be able to say, not only are you at risk of cardiovascular disease, but you're at really high risk in the next year or two.
Okay, so
We
there
need
is
to
a
intervene.
time factor there. There, you could say it.
Yeah,
so you're not
so
just
there
genuinely
was a
saying.
point to that long
Yeah.
anecdote.
No. So, I mean, it's almost, I don't know if people remember the movie Minority Report, where you could predict, who was going to commit a crime, at some point in year in advance. Giving people that information is very helpful in terms of heart disease, because there are things you can do to mitigate that, right? To mitigate. With breast cancer, there was the MammaPrint, which was another signature that showed people that they had a higher risk of breast cancer, or that it might respond to a kind of treatment, whereas another breast cancer would not.
Sure.
Are we at this point where we're going to see signatures for lots of diseases? And what about those diseases where there's nothing we can do about it? And there's actually a case with ALS now, where there's a very good predictive marker for ALS, but we don't have anything to do to treat that.
Yeah, I mean, look, that's always the challenge. You will often understand the disease before you have a therapeutic, but that understanding
right
is the first step. and many patients will want to know what their prognosis is in any event, or what their risk is, so that they can arrange their life accordingly.
You mentioned this small interfering RNAs. When you think about the sort of old world of drug development, it was small molecule, small molecule, small molecule. Then we sort of introduced the monoclonal antibodies. We've got ADCs, we've got, antibody drug conjugates, we've got proteins, we've got peptides, you've got something called BiTEs, which are bispecific T cell engagers. Is that right?
That's right.
So how, which of these arsenals, are we moving to a point where the specificity is the key, where you're sort of engaging only with the target, or as much as you can with the target, and not sort of a promiscuous amount of other targets in the body?
Sure.
Is that?
Yeah, no, I mean, molecular engineering, the goal, is fairly simple. You want to engage the target and create the effect that you want to affect. What you want to do, you typically antagonizing, turning off, or agonizing, turning on. And you don't want to affect anything else.
Right.
In other words, you don't want off-target effects. So, the new technologies and these new modalities that we're talking about allow us a much potentially greater degree of precision. You mentioned our bispecific T cell engagers. We've got a couple approved now, including one that targets DLL3, which is present on small cell lung cancer cells. A disease where there's been very little advance in decades in therapy, in a large phase 2 trial showed pretty remarkable efficacy, is now in a suite of phase 3 trials, and I believe has a chance to really transform the treatment of that disease. But these are molecules that don't have a single action, as you pointed out, but they are what we call multi-specific. A BiTE molecule has two hands. One hand reaches out and grabs a target on a tumor cell. The other hand reaches out, grabs a target on a T cell, brings them in proximity, leads to T cell activation and killing of the tumor cell. One of our beliefs is that we are now also on the threshold of a multi-specific world, where many drugs will be engineered to have this, more than one business end, and they're essentially creating new biology. We're bringing that T cell and activating it when it wasn't doing it in the resting state. When
you have all of these new exciting molecules, the drug industry, the pharma industry, has been subject to sort of big hype cycles, where people get very excited. I know you guys have a GLP-1 inhibitor that got a bunch of excitement in the phase 2 trial. Yesterday, Amgen's stock dropped 7% because an analyst had discovered that they're in a hidden tab in one of the early phase studies. There was some reduction in bone mineral density. That's a giant drop in the stock in one day. And are people sort of getting so wrapped up in the day-to-day of these discovery? Because it used to be that discovery took a long time, and now we're announcing everything and all getting very excited over each new phase that comes out and the readouts. And we're sort of almost setting ourselves up for these, reality comes to bite us moments,
Yeah, sure. Well, to speak to the release and the analyst note, we actually put out a statement this morning. We're very convinced of our interpretation of the data. We'll be going on record in a few hours.
Should we reschedule our conversation programming note,
at an investors conference out in the Los Angeles area, and anyone who's interested can get the details there. But I think you bring up an important point. We live in a world where science still takes time. It takes time to understand things. And, one of my jobs is keeping my teams focused and filtering out the noise, because the vast majority of what you will hear on any given day or read in the newspapers is noise. not signal.
Right.
And so, the key is, true north. What do you know? What do you need to know? And get
Right.
on with it.
Do we have time for a question, Ed? Or yeah. So we have any questions from the audience? Okay. Well, if so, we've got two mics on either side. Please feel free to stand up. We've got somebody walking down right now. Yeah. Just introduce yourself so that we all know. Yeah.
Is that on? Okay. Matt Andriotta, DNA Nexus. I want to go back to your point about the hinge moment. This may be just like a simple answer, yes or no, but like with all of these different collaborations, in your opinion, what are potential roadblocks for either acceleration or deceleration of innovation and interaction between these industries? You kind of touched on it just now, where some have to be a little bit more patient while others have that acceleration. So what do you see a potential strategy for that?
The biggest risk of this moment in time is actually the hype cycle. And the trough of disillusionment is coming soon. And we actually view that as an opportunity.
Isn't that a Lord of the Rings, the trough of disillusionment? Yeah.
The risk is that you can do a lot of really cool things. What does that translate into in academia or a company? You can do a couple hundred pilot projects that are really interesting, but they never scale and they never really move the needle. So, 80% of my job is keeping us focused on the dozen or 15 things that are going to not only, change our company on behalf of our patients, but also move the field forward if we get it right. And I think if you take that approach and let the noise kind of move past you every day, that is the way to success. What's the use case? What's the problem statement? What's the technology that you're going to bring to bear and then solve the problem?
Great. Thank you.
We have another question right behind. Just remember to introduce yourself.
Hi, this is Katya from LEK Consulting. So you mentioned some proteomic risk scores that you're validating in trials with 100 or so proteins. Just curious what technology you're using for that and what advances you think are needed to make those types of risk scores scalable and accessible for large numbers of patients.
Yeah, I mean, all you need is a simple blood draw. So that's the sample. The proteomic risk score was generated using a machine learning algorithm. And now, as I said, we're going on to replicate and validate that. But we're actually pretty confident that those data will hold up over time. But that's the basic technology.
How many particular proteins are you looking at in that signature?
We're looking at 5,000 to 7,000 in total. The signature is roughly 100 or so.
And in terms of the different populations, how consistent is it across race and gender and geographic?
Yeah, so that's part of the validation.
That's the validation.
That's
process.
part of the validation, right?
Yeah. Well, thank you, David Reese. Fantastic conversation, and we look forward to seeing what Amgen comes up with next.
Thank you.
We are very pleased to continue this conversation. I'm very pleased to be sitting in this chair and in welcoming Jacob Tyson. Jacob is the Chief Executive Officer of Illumina, one of the global leaders or maybe the global leader, if I'm probably, we'll see what Tatum tells me, in terms of gene sequencing and microarray technologies. And Jacob was at Agilent, a top executive there, and before that at DACO, which I'm going to mention in a minute, and actually started his own company, creating these cantilever sensors that are probably being used for very nefarious purposes now, war and and other things like that. But welcome, Jacob.
Thank you.
Jacob, I want to bring up DACO, which is a Danish company, which had been the partner for Genentech in developing a diagnostic for HER2, which was approved at the same time in 1998 with Herceptin.
Correct.
And so, as with David, you were sort of birthed in this incubator of personalized medicine. It's sort of funny how the two of your careers both started with HER2 in some ways. I know you came after that, but having that lens, just let me ask, where do you see us now in terms of this great revolution?
Yeah, for sure. No, thanks, Cliff. And I came in a little bit later than when Darko and Genentech developed together the Herceptin and the HER2 drug, or the Herceptin drug. But what was the promise there was that this is the start of the revolution for personalized medicine. It turned out that that was the first indication, and it took quite a while before the field started to have momentum. And if you sit in a diagnostic company, you can probably think that maybe the pharma company was not that interested to have stratified patient segments to the level that, of course, the diagnostic companies have. But it was very clear after the years that I came into Darko is that we saw that pharma started to be very interested in it. And it turns out that we were able to build up actually a great companion diagnostic business where we work with the pharma companies to develop the next generations of drugs, of personal medicine with drugs. And I think there was a few learnings there. PD-L1 with Keytruda and Opdivo was one of the areas that I was deeply involved in. And there were a few things that became very important there. It was not enough just to show that you could stratify the patient. It was also very important that you could go out and address all laboratories could do this, not only here in the US, but also the rest of the world. So I remember sitting with conversations with some of our pharma partners and looking at how we could actually get this rolled out into Kazakhstan, for example, where we, from a diagnostic perspective, say, look, this is only maybe 100, 200 tests a year. And for pharma saying, yeah, that's 100 and 200 times $300,000 or whatever the treatment could be. So I think another learning there is that it's not enough just to understand what the marker is, but also have the ability to run those tests in many types of labs around the world. So I think that's where we are now.
And there's another learning that comes out of this case study with PD-L1, which is that one of those companies who was developing a PD-L1 inhibitor, developed the companion diagnostic. That's Merck and Keytruda, and the other decided not to do that at first, which is BMS and Opdivo. And we saw what happened was when you do stratify those patients and find out who can really benefit from the drug, you get much better results and much better sales.
Yeah.
I
think I don't know whether we have anybody from Merck or from BMS here, so they can certainly go much deeper into.
We screened first before I asked that question.
But it is very clear that if you look at the progression of Opdivo versus Keytruda, is that Opdivo had a head start and I think had a very strong uptake in the beginning. But then when results came out on actually the efficacy of the drug, because you have stratified the patients even more clearly with Keytruda, it started to take off. So at least from a diagnostic company, that is what we want to see, obviously. Right?
And yet it's still counterintuitive, in this sort of, the pharma model used to be, you've got, you go for the blockbuster, you go for the biggest possible audience for your drug. And then we introduced this idea of personalized medicine, whereas we only really want to give that drug to someone who's going to benefit from it. But that's almost counter to the instinct for many pharma executives in terms of, trying to increase the sort of revenue for their, very big investment. But how do we, the Keytruda is a good example of how using the PMC approach works. But how do we convince others of that?
Well, I think there's a big opportunity actually to think about the drug and of course the personalized medicine in a very different way, instead of categorizing your drugs based on, if you look into cancer, you have lung cancer, and thereby you are being, you can go with Keytruda and you could go with a few other drugs. Maybe you start to see it shift over to look at actually either it's a mutation-based logic, saying if today, BRCA is, of course, has been very associated, of course, with breast cancer, but in reality, BRCA pops up in many, many different types
Sure.
of cancers. So instead of looking at the cancer origin, you actually look at the say mutation of origin and then define your treatment based on that. And I think that's where we are heading. For some type of diseases, but again, I'm not the expert. I know the pharma companies are the experts here, but I think that's where we see that's the setting, and that's why also from a companion diagnostic perspective, the thing about that is that you need to be as broad as possible. Going for right now, of course, back in the, we've talked about the Keytruda and the PD-L1, that's a one marker. But of course, now I'm sitting in a sequencing company. I actually believe that going to whole genome sequencing, looking at everything is going to be the future, because you don't want, as a patient or as a physician, to be locked into maybe a few panels that you think was associated with that cancer type, when we now know that there is much more information that can be extracted from the whole genome that could actually give you a much stronger treatment plan. However, it comes with a lot of other challenges and how to interpret and how do we make those decisions.
Yeah, I mean, you're absolutely right in terms of like looking at these signaling pathways and how they are, they're no longer site specific as we used to think of them. I mean, my daughter published a paper on HER2 in colorectal cancer. So it's very, this is the new reality. In terms of sequencing, and of course you do run the sort of largest sequencing company, we think about Moore's law. In 2007, I think the whole genome sequencing costs $1 million. It's now less than 600 or so. You tell me what the latest price is, but that is a dramatic decline in terms of the cost, and yet there's still a lot of opportunity that we've left on the table. Yeah. Why is that? With the cost coming way, way down, and we know the potential benefit of this in terms of finding actionable targets to treat, why is there not as much uptake as we would imagine?
Yeah, that's a good question. There should be much more uptake on that. But I think there's a few things that to keep in mind there is that.
We've got some time, so feel free.
So I think looking at the cost itself, I think it's been impressive to see what Illumina have been doing over the last decades on, as you said, driving down the cost of sequencing. And again, reminder, I mean, because sequencing was the main cost driver for doing sequencing. And thereby that became the number one element that you had to drive down from, as you say, millions of dollars, hundreds of thousands of dollars, now down in actually you can sequence the whole genome for, at least from a sequence cost perspective, $200. But now it also gets into a situation where the cost of the sequencing part of itself is not the main cost driver for actually doing the diagnosis. There's many other things into it. Sample prep itself, but bioinformatics, the interpretation, the time that it takes to interpret is going to be, and also there's still, if you look at the whole genome, you might be able to get out and say, look, you don't have the BRCA gene mutations, you don't have maybe HER2 alterations, but there are 5 other mutations that I actually don't, as a physician, don't know what to do with, and then there might be another 100 variants of unknown significance that I actually do not know at all what to do with. So that gives a challenge both for the physician to make an accurate decision, because you actually get overload of information. And I think that starts to be a challenge for right in the communities right now. And even if you look at it, I mean, I think you can actually take I'm making this up a little bit, but if you take a radius of 20 miles out of the big metropolitans, you have community hospitals there, you have community oncologists that is uncomfortable with that information and what treatments you're going to take. And as I spoke with Harvard yesterday also, it's not only about the treatment, but also the side effects of those treatments, that you're used to a certain type because of the standard of care was chemo, and you were used to a certain type of side effects. Now you're actually faced with a lot of other types of side effects because there's a different types of treatment that you've not been used to, and you might not have capabilities. So you end up in a situation where some of those hospital setting has to refer those patients out of their own hospitals, which in itself is might not be what they want to do. So all these things are probably a cascade of challenges that we still are facing.
Yeah, I mean, I want to come back to that cultural barrier, the cultural barriers in a second. I want to return though to the word overload, which you mentioned, and which is so significant now. And I'm going to ask the question I asked David about we're moving from genomics to all of these other omics, and looking at post-translational changes, modifications in the genome,
Yeah.
all of these issues where how do we sort of process all of this information now that we've gotten the cost of genome sequencing down?
Yeah.
We've got all this other stuff to deal with.
Yeah, so I think clearly that there's a lot going on right now and we're doing a lot of things also to simplify interpretation. So there's a lot you can do there. But I think the next challenge is that, and what Dave was talking about, that hinge moment, is that there's a lot going on right now, meaning that we are moving from, as you mentioned also, okay, after we sequenced the first genome, we all thought, okay, now we understand that. Turns out we still don't understand the genome completely and the impact that the genome has. We understand driver mutations, but we might not understand the complete pathway in the signaling pathway from what that driver mutation do. So there's opportunities now to use CRISPR technology combined with high billions of cells that use each cells as one experiment, use CRISPR to alter one mutation at a time and see the phenotypic outcome of each cell. So you can really understand, get to a much deeper level of understanding.
You're talking about looking at a single cell?
Yeah, so you use the same, so you if you want to look at a specific disease types, you understand the driver gene, you kind of understand on the highest level, but you don't understand the complete pathway, signaling pathway. You can now, with a single cell, you can go in and use CRISPR to mutate, one base pair in the genome, and you can then run the experiment in that cell, and then see, of course, the outcome phenotypically from the cell. And you can do that, do a mutation, one mutation at a time, or combined mutation, and so on. So you can do millions or billions of cells of experiment, and then you can read out all that out and get a much deeper understanding on what's actually happening. But the amount of information that is creating there is substantial. And I think the point is that, and now you can add, as David also talked about, proteomic information on top of that, epigenetic information on top of that. I think, as we talked about, the first part, when first we sequence the whole human, the first human, the understanding we have of biology has changed substantially. I think we're onto that again over the next 10 years with all the new multi-omic approaches we have now. It will change biology to the better. But what we also know is what we think is simple is just going to be much more complicated. There won't be one answer to anything. It will be, I think the future will be, it's not enough to do genetic testing. It's not enough to do histology testing. It's not enough to do, you need to combine proteomics with your spatial, with your DNA, genomic information, and probably much more. And it's going to be a great opportunity for companies like Illumina, but a challenge for the community on how we actually do this together.
The history of medicine is really sort of a history of these stepwise innovations, very reductionist. We figure out, okay, this cell is going to, or this protein is going to signal this protein, is going to signal this protein, and we're going to sort of look at that pathway and figure it out. In a disease like pancreatic cancer or many of the other advanced cancers, it's a melange of genetic destruction. There is so much mutational burden there that is happening. How do we approach that with a new lens, saying not looking for, say, one driver mutation where there may not be, but looking at the whole and seeing if there's another approach, either through the epigenome or transcriptome or proteome?
Yeah, I think that's exactly the point, is that we need to expand our focal point here in the lens to look at many other omics. And then the challenge is going to, how do you combine all this together? But I think the point is that, as you're saying, is that these diseases are much more complicated than looking at one modality. So the challenge is that how do you actually, so the first point we need to do is to start to do research on that. Having, and now we also, I think Dave was referring to that, is when he's looking at 5 to now we can look at 10,000 proteins actually using sequencing readout. You can look at epigenetic information. We're coming out with a simple way of looking at epigenetic. Today, it's way too difficult to look at epigenetic information, so we need to simplify that. So actually, researchers can start to bring that into their, into their research. So I think there's both a technology challenge that we are now overcoming, and now we will provide those capabilities or those technologies to the research communities, the academic communities. But on top of that, it's not enough just to do it. You also have to informatics platform that can actually help look at all this together. And all that it's happening now. And that I think is exactly with that hinge moment that David is gonna put trademarks around this, that it's happening, and it will change substantially for all of us over the next
speaking of which, I mean, you had said that in the past, a research scientist might spend 80% at the bench and 20% on the computer, and that's going to be reversed now.
Yeah, that will be, I think that's true. And I think also that's what Jensen from NVIDIA has been talking about, is that I think that's where he sees a big opportunity in this space also, is that you're right. I mean, biology is going from be something you do in the lab to something you do a lot in front of the computer, because we can, as humans, simply not cope with all that information and the complexity of biology as it is.
So one of the sort of answers to complexity is bringing in the wisdom of the crowd. And this iPhone here, there are about 1.8 million apps that have been made for the iPhone, and I think 6 million of them are like TikTok or whatever. Yeah. But given that point, I mean, in the machines that you've got, these remarkable next generation sequencers and some of those even more advanced technologies that you have you thought about opening that up to app developers to sort of bring in plug and play systems that they can develop their own way of looking for something beyond just what the sort of sequence or variation will tell you?
Yeah, absolutely, Cliff. I think that's, first of all, some of the reasons that Illumina has been as successful as possible. There are companies also in this audience that have been built on top of Illumina's technologies, and I think that's a big part of the success of not only Illumina as a company, but the ecosystem we've been a part of. But I think that's going to be even more important going forward, is that here in Illumina, we can help build up, we can bring out the solutions for, the sequences, we can do the whole workflows, and we can also come out with some informatics, but we can certainly not, as we talked about, this is going to be very complex, and we need to help the ecosystem, and we're very open for that. We know we can't solve all the problems, so really make sure we have it's probably, even though it's a good analogy, it's a little more complicated. Biology is a little more complicated than one and zeros. Right. So it's going to be a little more complicated than just saying, okay, we open up for different apps. But you're right, that is in principle what we want to do, because we cannot solve this individually, companies. We have to work together, both as tool providers, but also together with pharma and of course the rest of the industry, the healthcare industry as such.
So I want to talk, I'm going to use Ed's word here, ecosphere, which I really love. But I want to get people, if they want to stand up and ask a question, to sort of line up if you've got a question. Okay, we're going to people while we're waiting for this. Yeah. I want to ask you about some of the cultural barriers in that ecosphere. You've said we can't do this alone, any of this, but this whole revolution needs partners. It needs collaborations. It needs collaborations at scale. How do we bring that to bear?
Yeah, I think there's so many challenges to address here. There's both the technology challenge, the tools in itself. I think the industry we have shown that we can do that. I think now the challenge is that how do you actually get it out there? Reimbursement in itself, getting understanding from the payers, if you look into the payer systems today, actually reimbursement on sophisticated tests is I think some of the companies that are sitting here knows that the ASPs that they are receiving is far away from actually the reimbursement itself, levels itself. So there's still significant challenges in convincing payers about that this is the right thing to do. And of course, the challenge is that if you in US where you have a system where an average individual is sitting or have a lifetime with an insurance company of 3 or 4 years, it's of course very difficult to understand why you want to make decisions or give them a diagnostics today that actually have impact maybe on your health 5 or 10 years down the road. That I think is going to be a challenge for US, and you have to look into the single payer system around the world that's probably going to be better in implementing some of those treatment decisions strategies instead of what we're doing here. And I think that's something that at least here from a US perspective, we need to take very seriously. And I know, I mean, we're working with sovereigns around the world that is far ahead of where you think about also data, looking at that newborn screening, looking at the genome is following all the way through, but also making decisions early that is impacting your quality of life for 10, 20 years down the road. If you really want to do that, we need to think about healthcare in a very, very different way than just think about that you had 3 years on an insurance plan.
Yeah. All right. We've please identify yourself. Yeah.
Hi. In 2017, when Matt Gordon took over NIMH and from Judith Rappaport, I believe, I lucked into seeing their lecture. And Matt taught us that we go from DNA to cell to signal to systems, and then we have the phenotype. And that and it looks like we're bumping up against the limits of DNA in your work, which I followed a long time. So what's happening with signaling? I'm glad to hear you use that term. And can that be, and I don't know those terms either, of the physics of our biology, you know, the pluses and minuses, that also relates us to our epigenome, our sociome, our biome, and all the interactions, chemical and physical. Are we going there? Do you know?
Not really. And that I think, I mean, I don't know as a person, but I have an instinct that's saying that our understanding on biology is going to change. So the simple way of looking at a genome, transcriptome, and proteome, and so on and so forth, is probably going to change substantially over the next period of time as we learn more that this is a much more complex system that also, you know, if you look at the spatial information, we know that if you have different markers that is close to each other actually have a different impact than if they sit far away from each other. So the understanding of the system is becoming much more important than the understanding on the individual component. And you can't, it's not additive in the way that you just have the components and you understand the system. You need to understand the system and the components, and all this gives insights, but individually it's not going to be enough. So I think that will require a very different way we think about you know, the whole system in itself. But I think it's also important to say that doesn't mean that you don't want to do genomic testing anymore. You don't want to, and you're only going to do another type of information. I think it's going to be the combination of all of that is creating deep insight. But that's also why AI is starting to be important. It's not going to solve all the problems, but our human brain simply can't comprehend this. And you need data models to really help getting us to the next level.
Thank you for your question. We got just a few minutes left. Please identify yourself.
Matt Andriotta, DNA Nexus. I'm back. Quick, sorry about that. Quick question. You mentioned a lot of different specialties are needed. A lot of new data is generated. That means clinicians, bioinformaticians, scientists. What are strategies about creating a space for those people to collaborate effectively and drive those different insights? And then what would that be? What would make that an effective space for driving that innovation?
Yeah, and I think that's it's actually a good challenge. I think there's so many challenges we have to face. I mean, in the system here going forward and how we work together. And I can tell you that that's why I'm a big proponent of what we call the ecosystem, is that no single companies, whether it's in as a tool provider, as Illumina or pharma or healthcare system or health provider, can solve this. So bringing having an openness that this is not about saying if I do something, then somebody else is losing. This is all of us actually work to make the cake, so to say, much bigger. The opportunity is so much bigger, and we're just scratching the surface. And right now, the limitation is not the technology itself; it's how we actually build the ecosystem to get all the great brains together and actually open this up, because I can tell you there's so much more insights. We are happily opening up for our data generations, for the data we have, so people can actually build better algorithms on top of that. There's certainly need for standardization at some point of time, but you need to also let people get inspired before you kind of log into specific standardizations. But I think we need to do both. We both need to see what we can do and open up for all the good ideas. But then at some point in time, you have to log into some standardization. Otherwise, it's going to be the Wild West. Right?
We've got 1 minute left. I want to ask you one final question, Jacob. It's rare that we get a CEO for a biotech company who is a physicist. You trained in physics. You got your PhD in physics. From that perspective, we think in terms of when we think about chemistry and some of the other, even biology to some extent, we think, connecting one signal to another. In physics, it's a very emergent systems often are there. As a physicist, how do you sort of see the challenge that differently than someone else?
Well, I'm not sure I see that differently, but I think as a physicist, you feel like, of course, you've been trained in the foundation science, and thereby feel like, you have an opportunity. I think physicists believe thereby they should be able to understand many different of science, different science field. But I think what you really train in physics also is quantum mechanics, right? Yeah. And quantum mechanics is you learn very quickly that if you go beyond the hydrogen atom, you have no way of calculating in your hand. You have to do systems approach to it, and thereby you train very early in that it is much more complicated than just one marker. And thereby, I think you as a physicist understand that the ecosystem, the data, the compute power, and AI is important to really get this. The human brain simply can't understand.
So the takeaway is we need a systems approach. And with that, I want to thank Jacob Thijssen. Thank you.
Thank you very much.
Thank you very much, Jacob. We've built in a number of networking breaks, and the first one is going to occur now. We'll come back at 10:45 and look forward to a panel on investing in diagnostics. Especially for the admonition that we get deeper in the weeds in public policy, because PMC was founded on that basis. And it is in that context that it is my pleasure to introduce Brian Monroe, who will bestow this year's award for leadership in personalized medicine. And Brian was, as many of you know, the founder of PMC. The idea came from Brian when he was Vice President for Government Relations at Millennium Pharmaceuticals. So, Brian, it's a great pleasure to welcome you.
It is my pleasure and distinct honor to present the Personalized Medicine Coalition's annual award for leadership in personalized medicine to Emily Kramer-Golenkoff. Emily's story, there she is. Emily's story conjures up what makes the work of personalized medicine research painful and heart-wrenching and urgent. So let me say a few words about Emily first. I first met Emily as a young preteen when we lived in the suburbs of Philadelphia. While I knew she suffered from cystic fibrosis, never was there a brighter, more energetic, and precocious young lady, and her disease most certainly did not define her. She would go on to college and graduate school, all the while living with the specter of a terminal illness. One can imagine the paralyzing physical and mental manifestations of this reality. Emily has written poignantly about this, saying, "The hardest part of CF is the impact you have on the people you love. It's having to share bad news and being the source of their sadness and fear. It's clipping their dreams and ruining their plans. It's seeing their pain and knowing it's all because of you. So you have to ask yourself, amidst these challenges, what does a person do?" Well, let me share with you what Emily did. In 2011, she created Emily's Entourage. The nonprofit institution has since awarded more than $9.8 million in funding for research and drug development programs designed to develop life-saving treatments and cures for the last 10% of cystic fibrosis patients that will require personalized medicine treatments. Through its grant programs, philanthropic investments, clinical trial recruitment activities, and patient registry, the organization has brought potentially life-altering experimental therapies to hundreds of patients who would otherwise have few meaningful treatment options. Today, she has turned life-altering difficulty into a beacon of hope for her family, her fellow patient community, and I hope with all my heart for her. We've never given this award to a more deserving recipient. Emily's current situation prevented her from being here in person, but it's my pleasure to introduce Genevieve Mack, who will accept the award for Emily. Genevieve.
Thank you, Brian, and thank you all so much. I know Emily is incredibly humbled and incredibly honored for this award, and also heartbroken that she can't be here today. She's been in strict isolation since 2019 as we work towards treatment and a cure for the folks in the final 10%. She's going to share her remarks via video today, but I am truly honored to accept this on her behalf, and incredibly grateful for all the work for folks in this room, and specifically to Brian for recognizing all that Emily is and has done. So with that, please share Emily's words. Thank you.
Good morning, everyone. It's such a thrill and an honor to be here virtually with all of you today, to receive this award among a crowd of dreamers and disruptors, of visionary pioneers and change makers. I am so deeply humbled. I wish more than anything that I could be there in person with you today. I'm joining you virtually because I have been in strict isolation since March of 2020. With my advanced stage cystic fibrosis, catching a respiratory virus could be catastrophic, and I'm too hopeful about the advances in the pipeline to take any chances. You do what you have to do, and so I join you virtually. I want to say a special thank you to the Personalized Medicine Coalition Board, and especially to Brian Monroe, Ed Abrams, and Jay Wigglemuth, as well as the whole PMC team for this most special award. At Emily's Entourage, we are driven every day and with every breath we take by a singular goal and by what hangs in the balance. We do what we do because of the future we envision and that we believe our community so deeply deserves. Moments like this allow us to take a step back to see our impact and celebrate our progress. They energize us in ways I can't do justice with words. So suffice it to say, I accept this award on behalf of our most mighty entourage with overwhelming gratitude, pride, and invigoration. It is an honor of the highest order. As you heard, I am the co-founder of Emily's Entourage, an innovative 501c3 organization that accelerates research and drug development for CF. Cystic fibrosis, or CF for short, is a progressive genetic disease that primarily affects the lungs and digestive system. It causes thick, sticky mucus to build up in the lungs, leading to life-threatening lung infections. And the crazy thing about CF is that it's an invisible monster. Just looking at me, you would probably never guess that I have only 30% lung function, that I take over 30 pills a day, have CF-related diabetes and kidney issues, and spend roughly 4 to 5 hours a day doing breathing treatments and airway clearance just to stay alive. And that's when I'm healthy. When I'm sick, I go into the hospital for weeks-long courses of IV antibiotics. There are no breaks and no days off. And yet, if discipline with pills and treatments was all it took to stay stable, surely overachiever me wouldn't find myself dangling on the verge of end-stage disease. This monster of a disease has a mind of its own, and it ravages my body despite my hardest attempts to stop it. Everything changed in the CF world in October 2019 when a revolutionary mutation-targeted CFTR modulator therapy was approved by the FDA for 90% of the CF population who share one copy of the most common CF genetic mutation. This new drug is a true lifesaver. It's taken people off transplant lists, let them run marathons, start families, and dream of a future that they can actually believe in. It's the very pinnacle of medical progress for 90% of the CF population. But for me and others like me, we have rare mutations of CF. We belong to the outlying 10%. None of the breakthroughs are for us. We are the orphans of this orphan disease, still contending with the same killer disease that CF has always been, as our friends get their lifesaver, and we are the ones left behind. We are still waiting, yearning, gasping, pleading for something, for anything, so we can hold on. Our hearts are happy for our CF friends, but they're desperate too, desperate to join them. So back in 2011, I experienced the collision of two powerful and opposing forces. The first was that for the first time in CF history, there was real hope. CFTR modulators were in development that were poised to revolutionize the face and the trajectory of this disease. And the second was desperation. Despite my best efforts, my disease was rapidly progressing. My lung function hit the low 30s, and I was becoming resistant to all antibiotics, running out of conventional treatment options. The hopeful advances that were coming down the pike weren't going to be for people with rare mutations like me. And so it was that collision of hope and desperation that spurred the formation of Emily's Entourage. My family, friends, and I decided we couldn't just sit back and let nature take its course. And so we dove in head first, launching Emily's Entourage to speed life-saving breakthroughs and a cure for the final 10% of the CF community. At the time, game-changing advances were in development for one specific CF mutation, affecting roughly 4% of the CF population with the simplest defect to correct. And the groundswell of focus quickly shifted to the most common CF genetic mutation, given that it impacts 90% of the community. But for those of us with rare mutations, there was virtually nothing being done. The science was harder to crack, and researchers were singularly focused on the more common mutation. We were not on the research radar. Nobody was paying attention. And the worst part wasn't just the lack of treatments that were in the pipeline, but rather the lack of hope for future ones, as we quickly realized that there wasn't even an infrastructure in place to get new treatments targeting rare and nonsense mutations into the pipeline. And so we set out to create a foundation of the future that changes the way and the pace with which drug development is done. From the start, our mission at Emily's Entourage was singular: to bring life-saving treatments to the final 10% of the CF community, and to do it fast. We were not interested in recreating the wheel. Our goal was to fill critical areas of unmet need and to operate quickly. We sought to support work that otherwise wouldn't happen, or to make work happen faster than it otherwise would. Our strategy was as follows: to de-risk high-risk, high-reward research to generate early data and attract funding from larger organizations; to remove operational barriers by developing key research tools so that the only thing that stood between us and breakthroughs was pure science; to identify technologies and attract researchers and companies to CF from other disciplines to spur out-of-the-box ideas and black swan events; to infuse new capital into the CF space; and to cultivate a community of researchers, regulators, companies, people with CF families, and more, all focused on accelerating research and drug development for those in the final 10% of the CF community. This was all new to us. We had no idea what we were doing, but what we did have is a crystal clear vision of where we wanted to go. What we lacked in knowledge, we more than made up for with passion, laser focus, and tireless hard work. And it turns out that having an outsider status can actually be an asset to innovation. We leveraged our outsider status to see things in new and fresh ways and to reimagine the way research and drug development could be done. 13 years later, and we have come a long way. Among our highlights, we've raised over $15 million. We've awarded 38 research grants to top investigators around the world and secured over $52.3 million in follow-on funding. We deployed a venture philanthropy model to spin out a CF gene therapy company called Spirovant that is now in a phase 1/2 clinical trial. We've advanced 5 other projects that are now also at clinical stage. We've developed 37 bacteriophages that have been used to treat 34 people with drug-resistant infections, including 5 people with CF. And we've launched a patient database and a clinical trial matchmaking program to expedite clinical trial recruitment for this ultra-rare population. To date, we've supported recruitment for 5 clinical trials for mRNA therapy, gene therapy, an antimicrobial, and a novel aerosolized small molecule. These last few years, we've been racing, leading the charge to close the gap for the final 10% and making incredible progress. And while I may be the one up here on this virtual stage today, the truth is that I am accepting this award on the backs of an amazing community of people, of scientists and companies, people with CF and their families, donors and supporters, and so many more who heard the call, opened their hearts, and believed in our collective power to make a difference. Emily's Entourage is a testament to what is possible when a group of people band together and fight like crazy for a better future. And so accepting this award here today, it is about hope and action. It's about how change starts in the here and now. And it's about how we, Emily's Entourage and the Personalized Medicine Coalition community, we are leading this revolution, not only for those of us with rare mutations of CF, but for anyone with an illness or anyone who might eventually develop an illness or love someone who does. That was our motivation for starting Emily's Entourage 13 years ago. And that is my motivation for devoting my life and my waning time and breath to securing a better future for 100% of the CF community with no one left behind. That is my life's work. Because there are thousands of people just like me, vying to return to careers and to start families, to run marathons and explore the world, to plan for a future where they can live and dream unconstrained by the horrors of CF. They deserve better, and it's up to us to get there. We are well on our way thanks to remarkable support from so many, and we won't stop until we get there. My most heartfelt thank you to the Personalized Medicine Coalition community for this most purpose-affirming recognition. I'm energized, humbled, and overcome with profound gratitude. This means so, so, so much. So here is to fast breakthroughs and a future where we can finally breathe easier. Thank you.
So with that, we are on our lunch break. The staff will direct you to lunch upstairs. And you will pass signs for our sponsored session in room 217, Transforming Standard of Care Imaging into Precision Diagnostics. I want you to know you don't have to choose between lunch and the session. You will be fed at the session. So if you want to attend that, feel free to sit in room 217 on your way up. There will be signs and you can eat there. So we'll see you all back at 1 o'clock. Well, I am glad to see that our lunch program is so successful. We are going to get started here. So if you could take your seats, please. It's about 1:10, so we will get started with our next session here. And it is my turn to introduce someone to our audience here. And I think I want to start by saying it. In many ways, storytelling is at the heart of PMC's mission, at least in my mind. We believe that the story of personalized medicine will end in better health and medicine for all people. And we're fortunate to have in our moderator somebody who is remarkable at telling stories. She is 30 years of experience working with CEOs and companies to tell their stories across healthcare, technology, and many other industries. She is Kathy Bloomgarden, and she is the CEO of Reuter Finn, the public relations and creative agency. So with that, I'll invite Kathy to come on up to the stage, and she'll be in discussion with Christy Shaw, the former CEO of Kite Pharma. So thank you, Kathy.
Can everyone hear? Yeah. Well, actually, Christy's the one to really honor here. Christy's been through so much in the pharma industry. We first met at Novartis, where she ultimately became head of the US operation. And then she went to Lilly and finally to Kite. And it's really turned around really activities and outcomes in all of those wonderful organizations. But we all know in the pharma industry that we see obstacles and really come across really difficult problems to manage every day, whether it's data that doesn't work out, whether it's regulatory problems and interactions that don't go our way, whether it's stock price declines. I mean, these are all things that everyone faces. What's unique about Christy is that every time she came across an obstacle, she turned it into a positive. So, how do you do that, Christy? I mean, that's really difficult to manage and to actually master. So how do you think about turning a problem into an opportunity?
So, can everybody hear me?
I don't think it's on. Yeah. Turn it up. Yeah. Yes.
How about now? Okay, great. Thank you very much. And I agree with the introductions of Kathy. She's a great storyteller, a great supporter, focused on patients and innovation, and is really a catalyst for the industry. It's a pleasure to have spent all this time with you over the years. So for me, it's interesting because I think back to growing up, and I moved every couple of years until I was in 5th grade, and then my parents are from a farm in Iowa, and I adapted to suburban life onto a farm, and I was always told by my father there are no excuses. There are no barriers. It's how do you solve the problem? And from a young age, it was always that way. And I know my sisters were also, under the same roof, but somehow I took took it to heart, perhaps more. And as my journey continued, I found less and less tolerance of myself for always raising the why can't you, why not? Here's why I can't do something. Here's why that's not going to work. Because isn't that the easiest thing? Somehow, when you're 5 years old, you see the world as your opportunity and your oyster. And somehow over the years to adulthood, we squelch everything and we know too much. And we think of all the reasons why we can't do something. And it's the most draining thing to me is to think about what we can't do versus what are the possibilities and what would have to be true for us to accomplish what we're going to accomplish. So then the question is, well, why do you do that? And for me, it also is about, and I say this to others, a North Star for me is asking myself, what would I most regret? And I had a couple of regrets in my life. And so learning from those regrets of not trying my hardest to do something that is actually solvable or could be solvable would be the biggest regret I would have. Or the biggest regret when I took time off with my sister, what would be the biggest regret I'd have? Not, oh my gosh, this obstacle is so hard, I don't think we can do it, and shy away from it. It's such a minor thing to me versus actually getting the right people together to solve the problem and do things like cure some patients of cancer.
So people, of course, we all know, and everyone in this room has experienced it, that the industry is very criticized and people feel of greedy pharma. If you listen to the Hill testimonies on, I don't know, whatever, insulin pricing or whatever, you'll feel this, lack of understanding of how people in the industry truly feel. And many people try to instill this focus on patients repeatedly throughout their organization. And at Novartis, I remember we were having a town hall, as one often does in any big company. And the CEO at the time was asking people to come up and tell their stories about what had brought them to the industry. Why did they feel so dedicated and so impassioned about the work they did? There were several hundred people in the room and of leadership. And of course, nobody came up, raised their hand. And then Christy came to the front, the first person out of those hundreds. She stood up and she said, I hope you don't mind my saying this, Christy, but she said, my mother died young of breast cancer. And if I had known what I know now, I could have done something to help her. And that's always stayed with me. So, you really stood out, Christy, in, an organization, a leadership organization. How were you able to keep that front and center, keep that in your mind in a way that, really projected you throughout your career in a very meaningful way?
It is true. That is one of the biggest regrets I had is in 1996, there's no internet, not a lot of education. My mom was in rural Iowa, and we just let her specialist, which was really a community oncologist, which isn't a specialist, it's a generalist for cancer. And she never got referred to a clinical trial. Had she gotten referred, the aromatase inhibitors were approved the same year she died, and they actually extended patients' lives and actually lived much, much longer, and she could have. So it was the biggest regret of my life not having done that. And that's what actually fueled me even more. I went into the industry because my mom was philanthropic and my dad was in business. And I'm like, this is a great marrying of the two. But it fueled me even more to think about those stories of wow, there are answers. It's not just what one person says. It does take a village. We do need teamwork. We should look at all opportunities and explore everything. And so learning from that regret actually is what propelled me then when I took time off to care for my sister. It was looking at all possible avenues. And then why I left Lilly to go to Kite was, how is it possible there's a potential cure for cancer and nobody's getting it? We can do this. It makes no sense. And so a lot of that grit is, people say you were so courageous to leave a big job to take care of your sister, but it actually wasn't courageous. It was a lot of regret that I had, that I wish I had done something differently.
Well, I think it's also inspiring people to be taking something which looks like a difficult journey. And when you said, I'm leaving Novartis, I'm going to take care of my sister, it was shock to the organization. I mean, you were experiencing it, but the rest of us were like just looking and saying, what? How is this possible? So, again, you've done that several times in your life. You've taken this personal, deeply ingrained personal, pain and made it something that you then learned from. So what did you take away from that year of spending time with your sister and going through this painful cancer journey that unfortunately didn't end up in her being able to survive?
The best headline when I left, it's funny, I never actually made it into any big business rags, but at that time, Fortune actually said something about me leaving my job. I'm like, wow, I never got accolades for my business success, but leaving and stepping down from a big job was news. So that was interesting. I thought that I was patient focused because of what had happened to my mom, and thought in my head I was so patient focused, but it wasn't actually until I took that year off and experienced firsthand what it's like to navigate the healthcare system in the US. And from little things like my sister was on, 15, 20 pills a day, and the pharmacy changes the color of the pill and she loses it because she thinks she has the wrong medication. Or the bills from, the insurance companies where you have to validate and write, and, millions of dollars. Or I took, she was the $1 million study patient, but she had to fight for every dollar while she's trying to fight for her life. And so that's why, taking that time off, it was just such an eye opener. And the doctor comes in and tells her next steps, and he leaves, and she says to me, I have no idea what he just said, but she's not going to stop him and ask him because he's such a busy man. And so all of those pieces really helped me when I came back, especially at Kite, to say, so many times, we had a shortage of generic chemotherapy in the United States, which you have to have in order to get cell therapy to save your life. And my team said to me, Christy, we're going to have a downward sales trend for the next 3 to 6 months because there's, there's no chemotherapy, the type that's needed for preconditioning. I said, so what are we doing about it? And the answer was, well, it's not our product. It's this other company's issue. Well, I said, and of course to my team, I said, all I can see is my sister laying in her bed, waiting for her cells, because she went through the process, and being told, I'm sorry, we have a shortage of a generic product, so you're not going to actually get your cells back, and we can't actually try to save your life. So you make your plans for your funeral. And so we did. The team, said to me first, they said, Christy, you never think we're patient focused. I said, no, you're patient focused, but you're not patient action oriented, because we're not here to sell our product. We're here to save lives. And they were able to talk to the FDA. They were able to get chemotherapy through Canada, through Europe, and we never missed a patient. Similar to the story as, you worked with me on during COVID, here's a pandemic. We have patient cells in LA and we have to get them to Europe. And, President Trump at the time grounded all air flight. Well, how are we going to get this life-saving therapy back? And it was actually, we didn't say, well, sorry, we can't. It was working with the airlines, getting on the top 10 medical list, getting the flights out. And then when we couldn't get the flights, because I remember one time we both talked about this, the Chicago, all 9 flights, were stopped, and we couldn't get the cells to Europe. We finally did, but then we started chartering jets. And do you think that actually, you think it's $400,000 a therapy, in Europe, much less? We were making no money, but we were saving lives. And that mission of saving lives, we actually were losing $1 billion a year when I started. We became profitable in 2 years. And it wasn't because we looked at the product and how much sales we can have. It's because we looked at the patients and what the needs were, and how do we get every patient access to the life-saving therapy they deserve.
Thank you. Thank you. Well, I must say that I have to also share that Christy herself called the head of the airlines, I think it was United, and said, I'm sorry, I really need your planes to be available for me. So, she rolled up her sleeves and got that granular, and also arranged for carriers to be at the borders from Italy to France because they couldn't cross the border. So we had to have several different drivers picking up the cells, and the coordination and logistics for doing that were really complex. So,
and you actually did an article, because we hired a gentleman who used to provide medical supplies to war-torn countries. And we hired him and he helped us navigate all of the borders in Europe with ground transportation, how to get through the closed borders.
Yes,
So
he created an app on his phone that said how long the line was at the border exit at a particular site so that he would go around, because the timing, of course, for the cells to get back was very, very tight. So that was an incredible story. But if we think about these ongoing problems, the pandemic was one moment in time, but then of course, the problem you referred to with your sister and your mom, the community oncologist, perhaps not feeling quite so comfortable referring patients to centers of excellence, discovering that was a roadblock, patients not knowing that they could ask for a second opinion, that was a roadblock, patients who were deemed too old, at 65 or whatever, because they didn't get reimbursement. So how did you continue to keep an eye on these potential emerging issues that were going to really slow the access that you could see could be possible for saving lives?
well, some of the dots that I had connected is when I took a year off with my sister, we stayed at Cancer Hope Lodge in Philadelphia. And one of the things that we noticed is we were the only ones from out of the city, and let alone out of state. And it was because of these financial barriers. People couldn't afford travel, food, and lodging. And then I mirrored that knowledge with my mother being in rural Iowa and never getting access to a clinical trial. And then now fast forward, I'm at Kite, and patients aren't being referred. The academic centers are giving a lot of talks, like Dana-Farber, MD Anderson, they did all these community talks about the therapy, yet no patients are coming in. They keep giving patients, therapies that'll give them a few more months of life, a few more months. And then every time, of course, you give a therapy and time goes on, you have less of a chance of success. When you are referred for these therapies, they're only given at the time at the key institutions. And so like I had learned as, I started out as a sales rep, I started out from the ground floor up, learning all of the operations. The biggest input obviously is what's happening with the patient, and who we can talk to in the industry is the customer. So really trying to understand, why are the community physicians not referring? And there were a lot of barriers to why. A biggest one is they felt like they were going to lose their patient, and they felt an obligation to the patient, but there's also, an incentive for them to keep the patient, misaligned incentives as we talk about. And so what we did is we basically, we paid to deploy people to call on community physicians who can't treat the patients with this therapy. It's a cost, negative. There's no making up on profits or sales there, but their whole job was to educate the community and connect them to the academic center and build a relationship where if they knew they were the quarterback and they sent them, that doctor would send them back afterwards. And stop telling the patient, well, it could work, but it's highly toxic. You're at end stage. You don't know if you're going to live, and you might not be able to spend time with your family, and it's 2 months away. I mean, by then the patient's like, no, always no. And so we were the only company that deployed that education in the community to actually help with that whole referral system. And coupled with that, my sister and I started a foundation on behalf of our mom and our late sister. She, of course, was part of it before she died. But a piece of that is all about this disparities. So we, at More Moments, More Memories, we provide financial aid for patients who need help with travel, meals, and lodging. And we have found that only focusing on that, over 50% of our patients are Black and Hispanic, and over 50% of them are actually pediatric. And it wasn't a targeted effort. It was really trying to remove those barriers to getting patients referred to the academic centers.
So many people think, you told the story of, you were profitable in 2 years. You were not first to market with CAR T. It was Novartis, where you of course were. You brought all the insight, but many people feel if they're not first to market, they're really at a disadvantage and will never be, or very difficult to achieve, market leader. You obviously didn't accept that and worked really hard with an approach that brought you to be number one, and today far outpaced what was the first to market. So how should people think about tackling that second to market position or third to market position? What's the approach that really works?
So for me, and maybe it's learning from my commercial background, that the more competitors there are in a marketplace, the bigger the market becomes. So it was very sad, actually, that Novartis and BMS couldn't manufacture, because we needed them in. This whole awareness thing in the community, the whole direct to patient, all of the funding that needs to happen for awareness and education for directly to patients is so that they can have choices. And I'm not talking about, oh, big DTC TV commercials, whatever, just, you know, ground roots, how do we educate people, medical affairs, et cetera. And so that whole piece of that overall collaboration about cell therapy and the cure was missing. And so when I first went, we didn't know that this was a problem. So when I first went to Kite, my mindset is we need everybody to cure cancer and save lives. And then it actually became a burden that they couldn't manufacture, because then it became a burden on us, and how fast can we make it, and how many, manufacturing sites can we put up, et cetera. But that's one piece of motivation of why you should go in, because if you just remain patient focused, competition isn't a big thing. You don't, look to your right and look to your left and see what are they doing and how do I keep up. You say, what is the goal and how do I do the best for the mission I possibly can? And then I'm going to win. It's just like, how did you become profitable? Because you do well by doing good, and you focus on the right things. And that laser focus of, it's not about me, it's not about the company, it's about what we're trying to do from a mission of helping patients.
So you decided now about a year ago, I guess, to step back from Kite and take a pause with your family again. Your son Christian is, growing up, and you wanted a little time with him. We were all heartbroken, and we're waiting for your next journey. But now you are stepping in to help with Kyverna, basically a startup looking at CAR-T in the autoimmune space. So how do you have the energy, the courage to start all over again, to go through a journey where really work has not yet been done, and CAR-T itself is very complex: logistics, manufacturing, quality control, collecting data, patient recruitment, et cetera. It's a long journey. Where are your resources? What is your vision for being able to do that once more?
So first, the part about, leaving Kite is, at the height of my career, it's probably the most fulfilling job I've ever had, of taking something that was only in the US with 60 sites to go to 22 countries, getting reimbursement, and 25,000 patients treated to date, which means how many lives of their family, loved ones, half of those patients are still alive with a one-time cure. And I thought to myself, my son is actually going to be starting high school. Things are going really well. And it's probably the right time for me, because I talk about all the time, like the foundation. How do we give more moments and more memories to people, treating them, with cancer? And that year I took off with my sister, I didn't know if I'd ever come back, but all of a sudden, my younger sister took over because she was a match donor and I wasn't, and I was back in the industry. And I thought, I'm not going to get this time with my son again. He gets his driver's license in a year at the time, and I'm missing out on everything in his life. And what is my biggest regret going to be? My biggest regret is going to be that I kept working and traveling. I was gone 6 weeks in a row in the first 6 weeks of 2023. And I said, nope, now is the right time for me to take this moment and make sure I prioritize what I preach, which is, the people that I care about, and that I spend that time with them. And so Kathy and I were talking, and I said, it's interesting. I'm so blessed that I had that time with him. But now that he got his license, he's gone 7:30 in the morning. He gets home at 7:30 at night. And I'm like, what am I going to be doing with my time? So one of the things I found is, what I thought through is, what am I best at? And, I've been asked, startups, large pharma companies. But the sweet spot for me is this operational piece, where I can really look at clinical data, see how we get it to patient, and try to fuel it. And Kyverna is one where the technology is similar to what I was doing at Kite. We were able to scale it. And the data coming from cell therapy in autoimmune, whether it's lupus, whether it's neurology, diseases like Celine Dion has, or so many different types of immunology problems that could be solved, that nobody thought could happen with cell therapy because it's so toxic. And so as I choose what I'm doing, and it just so happens, the CEO now is my former chief commercial officer, and kind of getting a little bit of the old gang back, that we can actually help harness what we already did and do it for so many more people in other areas. So that's something I thought, this is something only I can do, because only I had certain experiences and can help. And so that's kind of how I see the choices that I'm making, is how are the things I'm choosing something that only I might be able to help with, versus just doing something to do something.
Yeah,
be
and-
relevant.
Yeah, and this ongoing focus that you have, with really bringing patients into the office and to meet with people is really very notable. I mean, I remember the story at Kite where a patient walked in to introduce himself to you and said, "You didn't just save one life, you saved two because I had a baby afterwards." Yes, so that was,
forgot
and he
that
brought the
story.
baby actually to the
Oh my
office.
God,
Yeah,
I'm
yeah.
tears.
So if we think about, if we think about, think about the outlook, the industry's up for a bit of an uncertain time. You know, we have an administration change. You know, we have new tools, though new technology tools that are going to make perhaps productivity enhance better and faster development times. There's lots of promise and lots of challenges coming our way. So what do you see as, you know, the sort of the outlook for the pharma industry, both in terms of the positives and negatives that we should really be thinking about?
So it has, you know, somebody said to me, "This is the best time, Christy, you could have taken off because of all of this volatility and the downsizing and the squeezing." And I tend to do better when we're thinking about the possibilities, not when we're trying to, you know, make numbers for the street. Sorry, investors. But for me, I think if we could get everybody to focus on patients and innovation, that every stakeholder could win. And if we all aligned incentives on patient outcomes and everybody was actually incentivized by patient outcomes, I think everybody would do better and we'd have much better outcomes in the United States. I think today, especially in the C-suites of big pharma, there's a lot of focus on profitability, and profitability does not equal innovation. And the investors aren't always holding that C-suite accountable for innovation. They're only holding them accountable for the quarterly dividend to make sure they're going to get those for how many quarters. And I would love the investors, those same investors, to actually ask the question. I remember we went to our largest investor at my last company, and I sat quietly because, you know, we were a small part of the bigger organization, and there were a lot of questions about dividends and, you know, quarterly earnings, et cetera. And at the very end, and it happened twice that day at JP Morgan, the investor said, "And by the way, Christy, thank you. That's all we want to say to you. Thank you, because we actually never believed in Kite. We never believed you could do it, and egg on our face, but thank you very much." And if I felt like if that kind of energy that my team had on patients actually went into the C-suites of big pharma, what a different world this could possibly be. And I'm not saying it's the C-suite's fault. I'm saying what they're actually held accountable to for shareholders, et cetera. If we could change that dynamic, but have everybody get what they need, but get it in a way that actually is a positive for everybody.
Yeah, I think there's going to have to be some new thinking in terms of how you value therapies also when you have the one-time, it's a high cost, but over the course of a patient's life, actually it's less. Additionally, the structure of healthcare with middlemen as it's currently organized is also an issue that may be looked at in months ahead. So, you know, it's a great moment to think about coming back. Yeah, we're looking forward to your coming back. I don't know if we have time for a few questions. Chris, do we have time? Okay, if anybody has any questions about, you know, Chrissy's personal journey and when she's coming back to take over a major role in the industry, we're looking forward to that.
Hi, Tina Annunziata from the American Cancer Society. I wanted to find out a little bit more about your strategy for where you supplied housing, transportation, and food support for your patients. So we're kind of taking a three-pronged approach to improving rates of enrollment on clinical trials. We're doing it from a research standpoint, from a policy regulatory standpoint, and from a patient support standpoint. So my question is, who did you target and how did you sort of advertise or make that public knowledge that you were supporting those aspects? Did you go to the physicians? Did you go to the researchers or the patients directly? How did that work?
So interestingly, this all started from a small foundation in Iowa that my mom was went to grade school, 6th in her class. Her best friend became a lawyer. I was taking time off with my sister. I said, "Eli, I have this idea." She hooked me up with a Dubuque Community Foundation who has nothing to do with healthcare cancer. They actually do land grants and legacy. But through those relationships, they're like, "Yeah, you can be part of our umbrella nonprofit." So we started there. And at first, it was just clinic by clinic. What we found the most successful was going to the social worker, the people who actually have that intimate relationship with the patient and actually take the time to help look for other avenues for them. Because we started looking at who was referring the most patients to us, and it was just a few across the country, but they were referring all of them. And it's funny, this morning I was at the Family Reach collaboration. There's about, 20, 30 charities that all got together on how do we collaborate. Because the one thing that we ended up doing is, so we had 1 patient a month, then we got to 8 patients a month. And then the Dubuque Foundation said, this is not our way of working. You can't stay here anymore because you're getting too big. And so I had a conversation with Family Reach. Family Reach did the exact same thing we do for clinical trials. They do before you need a clinical trial. So basically they acquired my foundation for $1. We are now the clinical trial access program, which we still run the More Moments, More Memories. But it's a big question we had this morning was how do you actually get that diversity? And the financial barriers are one of the biggest. The second one is people assume that that minorities, especially the Black community, will say no, but they won't. That's so long gone. The generation of Tuskegee, et cetera, it's not present. We just don't ask because the physicians or healthcare HCPs are biased to not ask that group. And so we didn't actually target diversity. Like I said, we just targeted inequity in rural areas, et cetera. And we did in Iowa, the grassroots flyers to the community clinics, et cetera, and started growing it there. And now nationwide, we're all over the country, over 300 patients since we joined Family Reach the end of last year, and a lot more if we had a lot more donations. So we would love more donations if you guys are looking for charitable organizations. Every single dollar that we have gets spent within 6 weeks. We spend, $100,000 to $200,000 helping these patients, and then we run out of money. So the great news, by the way, is that like 65% of the patients that we give grants to are still alive. The first patient we ever treated is still alive, and she was stage 4 metastatic breast cancer in Iowa. And so these are life-saving. It's not just an experiment anymore. These are access to life-saving treatments. Sorry, it was a long answer.
Great. Thank you so much.
By the way, thanks for what you're doing at the American Cancer Society.
Thank you for your work so much. What barriers do you encounter from research with a capital R? Like, I don't know if I can communicate what I mean, but trying to have patients have access to their own data and not have it totally taken away from them, double-blinded and de-identified and so on, so that they can use it to keep themselves well, which is the ultimate goal of data collection and of knowing our own data. Have you worked around that, or do you have to?
Yeah, so when we're in the clinical trial space, one of the biggest things you try to do is do exactly what the FDA needs to get your product approved. And when you add other things to it, it could make the clinical trial go slower. And then that affects a lot of patients if you can't get it to market faster, et cetera. But I do appreciate on the research side, there's some people that have access to, that have financial means to get their genetics tested, to spend $12,000 a year being tracked every year on all of this data, to Foundation Medicine, to Grail, the company that helps. I think we're getting there, but I think this is this disparate system we have in the United States. I mean, an example would be, I never thought I'd see the day where actually Europe did better than we did.
Oh, yeah.
And now, for example, and you were talking about the cost of therapy and doing it differently. We actually had multiple countries in Europe where we did outcomes-based agreements. So we said, it's a high price, but we'll guarantee the outcome. And if the outcome isn't exactly like our clinical trials, then we will rebate you. We'll give you money back. And the opposite's true. If we do better, which we never thought we would, guess what? We did better. We did better than the clinical trials because they started to learn how to manage the safety.
Mm-hmm.
And because this type of therapy is in the hospital and it's pretty much exactly like it is in the real world, we had 2 countries that actually gave us money back, and that never happens. But they're able to track the whole system because it's all centralized. And I'm not saying we need to go to that, but the way we have it in the United States is, AI, all of the means that we have, there's just a lot of roadblocks. Why don't we have a system where we know where which institution treats what type of cancer the best outcomes. Why can't we publish that? Why won't anybody publish that? It exists, everybody knows, but people are afraid to because they don't want to call anybody out. Well, this is life and death. Maybe actually if you called somebody, if you actually put the top 10 best and that's it, then those other institutions will be learning and we can all get better together. But this collaboration piece and the misaligned incentives are, I think, the biggest barriers. But to your point, I'm not actually answering your questions except
Yes,
to say I
sir.
understand your
You
pain.
answered it perfectly.
Yeah. Thanks.
Thank you very much.
Thank
We
you,
join
everyone.
thanking our panelists. The focus of the Personalized Medicine Coalition is not only on the United States. We aim to help advance personalized medicine across the globe. And our next panel is going to be reflective of that. I want to call to the stage my friend Tony Andrew, who is the scientific director of the European I always get this wrong. The EATRIS. Tony will tell us what that is, and also introduce his panelists, one from the United States,
Yeah, thank you, Ed.
Okay.
Yeah, okay.
Friends.
Yeah, thank you. So welcome to this global session. And thank you, Joshua and Monica, for joining us. I think that this morning has been fascinating and very interesting. And actually, it's quite amazing because when I come to the US, our organization is based in Amsterdam because the European Infrastructure for Translational Medicine represents a network of about 150 research institutions that work in the field of translational medicine, and personalized medicine is one of our main focus, I would say. I keep thinking, oh my God, the discussions that we have here are exactly the same discussions that we have in Europe. So why don't we have this discussion together, right? Because maybe we can learn from each other. And I think this is a little bit the spirit of this half an hour together. This morning has been very inspiring, and I like a lot one of the statements that Emily made, because she said that the ambition is that the only gap between the reality of the patient and the solution is pure science. And Ed mentioned this morning that PMC occupies the space in between science and the patient, which means that we have the capacity of closing this gap, right? With our knowledge and with our willingness. And actually, I think that in this concept, or along this line, sharing experiences and having an open dialogue between Europe and the US is important, and also, of course, globally. Actually, Dr. Rees said this morning, something which is, for me, is the essence of everything. We are more similar than different, right? And indeed, we are more similar than different. So to have a discussion about similarities and differences, we have here two experts and committed personalized medicine believers that, because of the responsibilities, they have a privileged perspective of the reality of challenges and opportunities in their respective ecosystems. And I promise this is the last time I'm going to use in public the word ecosystem, because from now on, I'm going to adopt the word ecosphere. For one simple reason, because a system is something vague, difficult to define, and a sphere is something that you can put your feet on the ground, right? And this is what implementation is about. So thank you for that, Ed. I'm going to steal the word from you. So we have here with us today Joshua Denny. Joshua is the CEO of the All of Us research program at NIH. And he's going to tell us about the objectives of the program, with this special focus on diversity, right, which is the essence of the concept of personalized medicine. And also, coming from Europe, my friend and colleague Monica Frenzel from the French National Research Agency. She represents a very interesting initiative called the European Partnership for Personalized Medicine. But first of all, I would like to ask Joshua to tell us about a little bit about the program All of Us at NIH.
Well, thank you, Tony, and it's a real pleasure to be here. You mentioned being sold on personalized medicine. I imagine that is the entire room being sold on the benefits of what we can do with personalized and or precision medicine. You know, the All of Us research program is a goal program across the United States to enroll at least 1 million participants who will share detailed information about health, their health trajectories, electronic health record data, surveys, things like Fitbit data, as well as biospecimens for genomics and deeper other kinds of omics that we can imagine, to really capture the whole health journey and be a platform to enable research to happen really across the world. And as Tony said, we have a real focus on diversity. One of the things, as we all know, it's been left out from a lot of genetic research, is diversity. We think of diversity, maybe first we think of in genetics of genetic ancestry backgrounds, but really we think of diversity in that as well as many other perspectives. People who live in rural locations, people who are sexual gender minorities, people with disabilities, anything that can affect your healthcare trajectory, like access to care, all those kinds of things come into what we think of and we want to capture. Overall, 87% of our participants are diverse by some type of metric that would be underrepresented in biomedical research. If you look at just race and ethnicity, it's about 46% are diverse by race and ethnicity. And you know, that's in stark contrast to the more than 90% of genome-wide association studies that are done with a European genetic ancestral background. And so this is the foundation. And the foundation is having a partnership with participants that hopefully will last for decades, that enables research, things like recontact, returning information, returning value, trying to stimulate precision medicine through clinics across the United States, across all health stages and age ranges, through return of pharmacogenetic results, germline genetic results from the so-called ACMG 59 currently, to be expanded over time, as well as fostering that research environment and potential for recontact for genomically different clinical trials, for things like that that we could do over time. And I've said the word genomics a lot, but we're not just about the genome, of course. It's about social determinants of health. It's about the exposome. It's about all those other factors that we could build into that, the proteome type risk scores that we talked about this, or we heard about this morning. All those kinds of things we feel as we want to make available to the world through commercial and international partners as well.
Joshua, is diversity a challenge or an opportunity?
It is a challenge. Tuskegee was mentioned earlier. We deliberately have worked with the Voices of Our Fathers, which is the kids from the Tuskegee experiment. We have worked with Henrietta Lacks' family. We have worked with community partners across the country very deliberately to try to form those relationships, to hear about the important aspects of building those relationships. And you can't, you have to earn trust. You have to be trustworthy, and listen. And so before we did anything, we did community engagement studios, kind of like focus groups in 17 cities, 77 different engagement studios. We've done surveys. We've been engaged with tribal nations across the U.S. For the last 7 years, and 3 formal consultations and a lot of other working groups on reservations, centrally taking advantage of virtual, and a number of other committees. So it's just a lot of deliberate action to form those relationships and listen and then be responsive. How can we provide value to communities, both at an individual level and at a community level?
We'll come back to the challenge, but before that, I would like to ask Monica about this initiative that she represents, largely unknown in the US and also in some parts of Europe, the European Partnership of Personalized Medicine. Let us know about this concept.
Thanks a lot. Hello, everyone. It's a pleasure to be here. The world begins to be small when we are talking about personalized medicine, because the more we stratify, the more we are having smaller cohorts, the more we need to work together to make something reasonable out of research and bringing it to application and really to the benefit to the patient. As usual, I'm overwhelmed in these kind of meetings, so much input, so many aspects we have to work on. And I totally agree, we are all ambassadors to put things forward. But I think we are the ones also to know that there are difficulties. It is a mind shift. It is a different responsibilities. It's different knowledge we request from healthcare providers. We put the patient in another role. It means a clear change of a system. And I think this is what Europe tries to push through centralized programs like the European Partnership for Personalized Medicine, for example, or the International Consortium for Personalized Medicine that was already in contact with PMC. So it was great to have you recognized by IC Permit as initiative for the work you are doing. You participated in one of the conferences. We are starting to be connected. So the European Partnership for Personalized Medicine is bringing together mainly funders, ministries from health and research to push the personalized medicine agenda on different levels. The most important part, I think, that we are doing is pushing research on various fields, trying to get understanding where is an application field of personalized medicine. So we have a budget of 300 million euro for 10 years. So I think the big difference here is also trying to create an initiative that is running for a time where we can really move things and push and support also research. On the other hand, we notice that the big difficulty we face is the transition from research to practice. Innovation is not always market access. It is also thinking about the needs, the implementation questions. And I think here for Europe, it's really important to collaborate because we have different systems. We have different countries. We are organized on regional, national, regional level. And what we noticed is if we would like to make personalized medicine a reality, especially in Europe, but also worldwide, we have to collaborate. So by bringing the different partners together, we are pushing strategic and policy development to work on the different levels: education and training, knowledge sharing with the patient and the citizen, the researchers. How should research be created that research outcomes are ready to be at the moment implemented, because also they are meaningful. The research is from the very beginning on meaningful, including patients and citizens, for example. And then trying to, well, see how to cope with different regulations we have. We will not, in Europe, harmonize healthcare systems, but we can try to align them and to try to agree on common policies and maybe to make, in this way, personalized medicine accessible for everyone, and equal access, I think, is really an important aspect. So maybe I can stop here.
Yeah. Well, I think it's quite interesting, this statement that we will never be able to harmonize healthcare systems in Europe, right? I got the feeling that it's a little bit of a similar challenge in the US. I mean, this is not an homogeneous canvas, right? It's also diverse, and I don't know if to use the word fragmented, but
I use the word fragmented.
Okay.
So go ahead. We did realize yesterday in talking that there are maybe more similarities than we realized in the many different individual silos of healthcare that we have across the United States and what happens in Europe. While there may be national healthcare systems in some places, each country is different, and cross-border is obviously hard to share. So there are real distinct challenges. Now, one of the things that I was really proud of our teams to develop was the ability to pull electronic health record data together from many different institutions and harmonize into one common data model across a number of different EHR vendors. That was done in, I guess, first around 2018 by our program, and was taken advantage of in a big way for COVID. We saw how we could mobilize lots of data across country boundaries, and of course across all the US-based silos in COVID to solve what was a public health emergency. That made a lot of things easier in terms of regulation, but I would argue that it should be a call that we should be looking to continue to harmonize the kinds of data that we can make available, especially with participant consent. We have deep consent. We still, the vast majority of the people, more than 95%, agree to share their electronic health records data, but we are not able to gather it from that many people. We do get a lot of it, and we have a number of healthcare systems participating, but there's still a gap that we need to close. And I would argue it's probably similar as you look at Europe and across the world.
So you think that sharing data or sharing tools that facilitate the exploitation of data, which is maybe a slightly different approach, is an opportunity for creating this kind of global, let's say, community for tackling the big challenges of personalized medicine? Is this one of the ways to go? What do you guys think?
I think personalized medicine is based on data. I think that we are now like pioneers thinking we need to push personalized medicine is because the future is more and more data are available and we need to be ready to use them of different type, style, and well, to treat them. And I think what we also notice is general diseases are with the stratification potential. We heard a lot of biomarker, diagnostic tools. We will have more and more smaller populations of patients that need specific treatment. If we remain in our country, especially in Europe, but I think even US and China, they have comparable issues at the moment, and we will not have the successful treatment therapy afterwards. So yes, we have to work together. And I think we need to find, and there are good approaches on the way, how we can use data together without or in complying nevertheless with our data securities and with complying with the different consent forms. But again, this is a question that we need to prepare now. We need to prepare the baseline to have regulations in place that are allowing us to have the, first of all, the right consent form, to use the data, to mine the data, and to have a way to exchange them or to make them accessible at a certain level together. So I think really this is an important aspect to data and access to data.
We'll come back to this later, but I don't want to miss the opportunity now that you are here of asking you to do a little bit of a mini SWOT analysis of the strengths and the weaknesses of the personalized medicine ecosphere in your different, it's the same sphere, but different sides of the sphere. So Joshua, what would you say is the big strength and the big weakness of the personalized medicine development agenda in the US?
Glad you didn't ask me to do all four at once of the SWOT because I'd invariably forget something. You know, strength, we do have a lot of data. We have a willingness, and I think we're showing that we can bridge some of those challenges and hindrances we have with historical research on diverse populations. And we are being inclusive and people are joining the program. I think we are seeing in real time, you know, only about half of the people actually want genetic health results back. So that tells you something. It's a little different for different populations. So that tells you that we have a gap in understanding that we can address. Now, I have a strong bias. I got both of my sets of results back, but you know, I also have a strong bias that pharmacogenetics, there's really not stigmatization risk for that. And there's really not health insurance risk or life insurance risk, et cetera, with that kind of information. So why is the gap still there? So there's an understanding gap, and that's a weakness. And that's one of the things we definitely wanted to tackle head on. Another strength is we do have a strong healthcare system that can do a lot of good things in the United States. But, you know, its adoption of precision medicine, as we all know, is not where we would like it to be. One of the reasons I was so excited about being involved in the possibility for all of us, its inception was the idea of allowing anyone in the US to join and to get genetic results that go take to their doctor and have them try to figure out what to do with it. Because I thought if you had patients at the center of carrying that information forward and having the discussion and talking to other, you know, friends and family about what it looks like, you create a cascade effect of understanding, and you put patients at the center of it. And really not just picking, again, you know, sort of the well-educated, high socioeconomic, sort of well-off set of patients, but, you know, a really broad swath of patients. So those are a couple of the strengths. Obviously, tons of data we can generate, availability. A weakness that's a risk is we need huge numbers, right? And I don't view like we keep track of how many publications, et cetera, are coming out of our program, right? But really what you should be seeing is we should be, you know, in a non-competitive way, people should be using all the biobank and other resources together across the world together. And so they should not be using ours, they should be using us, UK Biobank and a corporate data set. And we need to figure out ways for all these sets to work together as good as possible. And as we have improved and created new data sets, we are risking more siloization. Again, And we got rid of some of that for a while and got more collaborative. And I think, we have to be careful not to reintroduce that too much and break down those silos.
Yeah. You think this is going to have a positive impact in generating trust in the public? Because at the end of the day, adoption of personalized medicine will largely depend on the perception that the public has, right?
Totally. I mean, we got to push towards, again, a good friend of mine, our chief engagement officer, talks about, the importance of, being trustworthy. Like, we just have to try to be clear, communicating, not over promising, being transparent, admitting when we're wrong, and getting, data back that they actually want. And being willing to go that extra mile to meet people where they are. And education's a big gap that we can tackle head on.
Do you think in Europe we have a similar journey, or you feel it's slightly different?
I think the big barrier, I always like to start with a problem and hopefully to start with more enthusiastic solutions or advantages. So the big issue, I think, and difficulty in Europe, and I forgot it in our discussions yesterday, is language. We have language barriers, and this is really different languages on a very small area. What is making it quite complex when it comes to communication. So as soon as we start to communicate to citizens and patients, it is hard to launch one single campaign. So that's maybe a big issue. And then in general, when it comes to the implementation and innovation, Europe is consisting, as I said at the beginning, of different countries that are organized in healthcare, but also education and training, even on national, regional level. That is making a system very complex. Market access is quite complex because everyone has its own regulation. So this seems from the outside very complex. But I think the good thing is Europe has understood that this is a challenge and they are working on it on different levels. We talked about data. There's a European health data space under development. We try to have common data regulations that are quite challenging since they were installed, but nevertheless, it helps to have this alignment. The different regions or, well, mini healthcare systems are also a very good advantage to create incubators, to have a small, how do you say it, ecosphere?
Ecosphere.
Thank you. Having
official term from now on. Huh?
Where stakeholders are very close. So you can try on a small system sphere to implement something. And this is the strength, I think, from Europe. Sharing knowledge, creating consortia that despite the real language barriers, they develop a common language nevertheless, that helps them to collaborate, to find a common goal, and to share knowledge, and to communicate about good practices, to be able to say, okay, we don't work everyone and we don't do everything in parallel. We waste money, I don't know, 40 times because everyone has to do it alone. No, we learn how someone was doing it, and we try to not redo the mistakes, and even we try to do things together and save resources in this way. I think this is really something that Europe can also bring to the world, despite differences, how to collaborate together.
I think this is very interesting because in this cultural change that we are talking about for adopting a personalized medicine agenda, one of the powerful drivers for a change at the cultural level is the concept of the story of success, right? Because this is really what people opens our eyes to say, okay, this is really possible. Okay, so collaboration is of course, is the magical word, and we have an ocean in between, right? But physical distance should not be a barrier for collaboration, and I see that there are many potential areas where let's say, the European personalized medicine community and the US or American one in general, I think there are several potential areas that we can do things together now. You mentioned about, for instance, or the possibility I understood in between lines of creating shared data platforms or aligning on regulatory processes. Could you please elaborate a little bit more about ideas of things that we can do together to generate trust and accelerate the adoption of personalized medicine, Joshua?
Well, I think you alluded to a couple of them. One is we should build some of our our infrastructures, our data sets with eye towards collaboration. So insofar as we can use common data models across our phenotypes, we've done a really good job with genotypes. If we can do a better job with phenotypes, and there are some standards that exist that would be helpful. Thinking about how you set up your infrastructures to allow for cross-analysis, whether it be federated or pooled, whether you have a trusted research environment that you can bring things into, figuring out those kinds of things would be a couple of them. And then just thinking about more in less of a competitive, but more of a cooperative space, I think is helpful. I think if we are looking forward, as we think about, we've had a few references to changes that may come in the next couple of years. And as we think about that, we have a mission to serve towards real people and real disease that is just such an important calling. And I think we will do better working together on common platforms. I mean, we desire to be a platform that hopefully other people can come into and partner with to be better than any of us could be individually. And I think that's just the way we have. Part of it's just thinking that way. How can we invest in something that will rise all boats instead of our own boat? Now, I obviously, sir, come from the federal government of the United States and see this as a public good, but I think that it can also be the good for many different avenues.
Monica?
Yeah, I also come more from the policy perspective. So I really see the advantages of aligning of regulations or trying to find common incentives, common way to develop policy strategies towards maybe comparable regulations, allowing also a better connection of the public and private sector. Still, I'm coming, as I said, more from the public ones. Still, I have the feeling if we talk about language barriers, there are still a lot. And so this is why it's so impressive to hear all the talks today. I think I'm also from the funders perspective, I think about bringing different communities together on different levels. The researchers, it starts there. Sharing of knowledge. I think our researchers are really the ones that are pushing the innovation part with good ideas. Then trying to bring public-private partnerships together to better understand how we can better support this innovation market access pathway. And then the policymakers, and we need support to train also our healthcare forces. So to learn how what do we have to change in terms of education and training for our healthcare forces, support tools, because they are not and they cannot be all specialists, but how to support them that we can implement personalized medicine. Yeah.
Thank you. I think we have a couple of minutes for questions. Please go to the microphones. Ed, yes, the creator of the ecosphere.
Thank you, Tony. I wanted to ask Europeans on the panel what the role of payers, what is the role of the payer in Europe to advance personalized medicine? Or put another way, is it an advantage to have a single payer system?
Well, I'm not the ones that is changing this system, but it is of course creating complexities, especially in Europe. Every country is organized differently in terms of reimbursement. But what I see, for example, in France, I think what the ministry tries to push is to support that new innovations are like put under practice to see if it's working. What is happening afterwards, also the reimbursement process. So there's a new mechanism, for example, in France currently in place for in vitro and companion diagnostics that would not reach the clinical application if they are not confirmed and with the legal authority, health authorities. But there's no the policy or the ministries that have understood that they need to push and to support this transition, so that after also payers they are agreeing to support a new innovation into implementation. So there is specific funding now available for new diagnostic tools to be tested through different experiences that could be supported, but also through clinical trials. So it is maybe a way to create the evidence that sometimes we need for the reimbursement part, right? But I don't think that in the next years we will change this discrepancies between how healthcare systems are developed and how reimbursement takes place, for example.
And the last question.
Oh, the last word. I was privileged to attend the celebration of the 100,000 Genomes Project in London with my son, and we were the only Americans in the room. And I signed up to come to this conference because you are coming together here right now to celebrate again, trying to make this a global effort because we all need this biology, this science, these doctors, and this care. And because I suggest that the common language is biology, and you in each separate country need only translate it from biology to children in school. So they will have the language of their own biology and be able to take their own bio-identities and use that to help themselves, to heal themselves. And I suggest that these children, these generations will solve the problems we're trying to define here. I guess that's all. I should have a question. I'm sorry. I guess I'm just saying thank you.
No, thank you. I think you just give us a boost of positive energy, which is the kind of thing that we need for keeping our passion alive. Thank you so much. And thank you to you for this fascinating.
Okay, so next up, next moderator is Felice Fryer. She is the president of the Association of Healthcare Journalists and has quite a bit of experience in healthcare reporting, and we think, including most recently at the Boston Globe. So we're really pleased to have her. We think she's the perfect person to moderate the next session, which is titled Early Detection and the Future of Cancer Care. So Felice and her panelists are welcome up.
Bruce? Oh, there he is. Okay, one more. All right, we are here to talk about a really exciting development in the area of cancer care, and that is that there are these blood tests that have been described as game changers. There's a colorectal cancer blood screening test that can detect 87% of colorectal cancers at early stages. There's another blood-based screening test that can identify patients who have early stage lung cancer. And yet another test looks for the presence of multiple types of cancer at once based on biomarkers circulating in the blood. So what does all this mean? We have some great distinguished guests who can tell us all about it. And their titles are all up here, but I feel like I should introduce them anyway. Dr. Cristina Annunziata, who is the Senior Vice President, easier for me to read here, at Extramurals Discovery Science of the American Cancer Society. Tom Baer, Chief Medical Officer of Multi-Cancer Early Detection at Exact Sciences, and Dr. Sapna, okay, Singhal, who is Director of Strategic Planning for Prevention and Early Cancer Detection at Dana-Farber Cancer Institute, and Bruce Ratner, who is the author of a book called Early Detection, Catching Cancer When It's Curable, which I think is available here, isn't it? If people, it is.
Free.
Oh, and it's free. Wow. So, so to start out, I actually would like to ask each of you, maybe starting with Tina, just going across, what does this mean for the future of cancer care that we have this type of detection available?
So for the future of cancer care, I think this gives us an opportunity to detect cancer early and stop it before it is a full-blown cancer. So that would really, really be a game changer for patients. I always tell my patients it's better not to get cancer than to get cancer. So really pushing the screening guidelines that have been put out by the American Cancer Society that does not yet include these blood tests, but we'll talk about that later. But if we could implement blood tests for everyone to prevent cancer or detect it when it's early, I mean, I think that would be a huge benefit to the population in the United States and globally.
Tom.
Sure. Just to add to that, I think when you think about the power of cancer early detection, a couple of good examples are cervical cancer. You know, a couple of years ago, I had the privilege of being invited to the launch of the American Cancer Society Roundtable on cervical cancer, where the stated goal was the eradication of cervical cancer, which is almost hard to imagine, but that's kind of where we are. And if you look at colorectal cancer, the age-adjusted mortality for colorectal cancer over the last 30 years has declined by 55%. And that is a cancer where screening is well established, although we still have 60 million screen-eligible individuals in this country alone that have not been screened or are behind on screening. But even with that gap, we see that robust early detection can make a real difference. I think as we look over the horizon, you know, my focus has been on multi-cancer early detection. And so what we're seeing is hopefully a future where these blood-based tests will extend the reach of screening to the many cancers that are currently left behind. About 2/3 of cancers we don't screen for today.
I think, you know, one of the analogies I've started to use when we speak about this is that we would never wait for somebody to have a heart attack to check their blood pressure and cholesterol, yet that's what we do for cancer. You know, our approach to cancer has been really reactive for the most part. You know, we wait for a cancer diagnosis, and that's where we start thinking about treatment. And I think this whole field is at a hinge point, you know, right now, because of the availability of all the novel technologies. So the blood-based markers, but there's CT-based screening for lung cancer. There's the implementation of existing standard tests. So at Dana-Farber, we really decided to jump fully into this and launch what we call the Centers for Early Cancer Detection and Interception. And we're building programs to identify people who are at increased risk of developing cancer, either through genetics, through their germline predisposition, through precursor conditions, or through the finding of a positive MSAT test, with the goal of bringing them in, implementing screening and surveillance programs, navigating them through the system so they get the test that they need, and help educate the physician workforce and the healthcare provider workforce about the benefits of screening. And so I think this is really, we've talked, this field, I feel like sometimes we focus a lot on the challenges. I think it's a really exciting time in the cancer diagnostics field, and the potential of early detection and prevention is really tremendous. So I think there's a lot more coming down the pipeline in the future.
Bruce.
So I wrote a book for a very simple reason, because I believe early detection is about the only way to really cure cancer. In metastatic, you do not cure cancer at all. And early detection is everything. That's why I did the book. Now let's talk about MCEDs. The problem with MCEDs is money. It takes a great deal of money and Thomas can talk about that, to be able to create a test that can test for 50 cancers and then implement. It's extremely expensive, and I'm worried that there's not enough money being put into MCEDs such that it's going to really come out in the way that we hope it will. And I must stress, I think it is the future of early detection, but I'm worried about whether or not there's the money for it. But one last comment is, let's not forget that 20 to 25% of cancer deaths are lung cancer. So if we just solve that one, we reduce cancer by 20% to 25%. And yet only 5% of people who are eligible to get lung cancer screened actually get screened. So we wrote the book, but we wrote the book in large part to create a platform to be able to come up with models that really work. And we're starting with lung cancer because that is the #1 killer. And we want to try and get something done and done quickly.
Tom, did you want to address that? You look like
The not enough money part? Well, I think innovation doesn't happen, or at least it doesn't succeed in a vacuum. The ecosystem and the conditions that lead to investment are really important. So for multi-cancer, there are 2 or 3 things that we could really use from the universe. One is a pathway to reimbursement if the tests are successful. I don't know if everybody knows, but the US Medicare system is built around diagnosis and treatment of disease. Preventive services require legislative action to create a benefit category, and that is a high priority for the field. We also need to agree on evidentiary standards that lead to adoption. What is the level of evidence needed for these tests to make it through FDA approval, but also to be recommended by guideline makers and adopted by payers? Because ultimately, that's the only way to make these tests available broadly. These are areas where we're not there yet. There's a lot of discussion and debate in the field. But I think to stimulate investment in this area, these conditions would be helpful. If we could address these things and have a clear pathway towards success, I think the universe would find enough money to invest in the development of these tests.
I sure hope you're right.
Yeah.
I'm not as optimistic, I have to say. And I know what you guys spend on marketing just for Cologuard. And you have 1,300 salesmen just to sell Cologuard. And so I work on implementation. And implementation is extremely expensive, extremely hard to do, and it takes a lot of money to do it. And we have a fractionalized healthcare system, which makes it even more difficult. So I would love MCEDs to happen. I am worried, I don't say they're not going to happen. I'm worried about the economics of it. Just to give you another sense of it, NCI spends $7 billion a year on cancer. Only 9% is on early detection or prevention. 9%. $600 million in total out of $7 billion. Keytruda, by the way, has $26 billion in revenue. So that gives you a sense of where things are at. And that's just plain wrong. That's a wrong emphasis, and that's where I come from.
Sabna, could you talk a little bit about what you just described, this early detection program that you set up there? How do these new technologies and these new tests, are they part of it at all, or is that down the road?
Yeah, I think that they're going to be baked into the program. And one of the things, and Tom and I have talked about this before, is that, are we going to have one test that fits everybody? That's the holy grail, as everybody would like. That's probably unlikely. And I think one of the things that we're thinking about is precision prevention, in the same way that we think of precision treatment, is that you really need to risk stratify your patient in front of you, understand their genetics, understand their risk factors, understand their family history, understand which precursors they have, and then tailor their prevention strategies. So we do have a clinic that's led by Betsy O'Donnell, who is going to evaluate patients who have MCED tests in the community, because we know, as we've heard already, that the primary care physician community is not going to know what to do with them and how to work them up. So I think we definitely need a resource, and we recognize that. So that's one of the clinical offerings. But as I said, we're actually building early detection programs and monitoring programs in every solid and liquid tumor program. So for lung cancer to get out in the community, just to let people know about the benefits of CT imaging and bring people in to screening. That's a component. Some of those people might get MSAT testing, but not all of them may need it. So it's a very tailored approach rather than a one size fits all approach.
So are you saying that the blood test would be, people would still be coming in for CT screening? This would not be a substitute for it?
I think these, and maybe Tom can comment on this, I think one of the big dangers of that physicians worry about for the MSAT testing is that of false reassurance. Just because you have a normal MSAT test doesn't mean necessarily that you don't need a colonoscopy. And we don't have the garden representative here, but I think it's a great advancement that we have a blood test for colorectal cancer, just because it's going to get more people screened. But that doesn't mean that if you have a symptom that requires you to get a colonoscopy, you should think you're safe from colorectal cancer. So the messaging and the education about this is really important. I don't think one screening test or one blood test is likely, it's not going to be better than mammography, for example. We have very good breast cancer screening tests. It's really important that the population and the healthcare providers understand that.
So how widely used are these tests right now?
For
Blood tests.
MSATs?
The blood tests, yeah.
You want to talk for them?
I mean, this is really a nascent field.
It's
so
not, they're really not out there.
they're just, I think this is a good time to have this discussion as we begin to move in that direction. But none of the blood tests are in widespread use, widely recommended. Our test is still in development. In the MSAT space, there's one test that I'm aware of that's commercially available, but is not widely reimbursed. So it's, this is an early field. And I do think, just to speak to that complementary nature, blood is a wonderful fluid to interrogate, particularly for multiple cancers, because it's kind of the ultimate integrator. But it's unlikely that it will compete with the sensitivity of tests that are directly evaluating an organ of interest. So, and colorectal cancer is a really good example. When we look at stage 1 colorectal cancer sensitivity or precancer sensitivity of the two tests that have published their results or presented their results, it's much lower than stool-based tests or colonoscopy. So those will be a really useful tool for individuals who are not willing or able to get screened with colonoscopy or stool-based tests, but they're not going to be a full substitute for the foreseeable future, just because particularly precancer just doesn't shed a lot of biomarkers into the bloodstream.
Well, as a citizen, I was kind of hoping for an alternative to colonoscopy, but that is not what we're looking at right now.
I think there are alternatives. I think Cologuard is an alternative. I think the Garden test is an alternative, but you have to understand the sensitivity, specificity of each of those tests and then make an educated decision. They're not as good at picking up precursors as they are at picking up cancers. And I think that's one of the limitations. But if you said to me, I'm not getting a colonoscopy, I don't care what you say, then I'd rather that you get one of those tests rather than anything at all. Nothing at all.
And one of the problems is the false positive rate is very high on Cologuard, for example. Am I right?
Well, I mean, I think the single cancer tests are typically geared towards sensitivity, and that's true of mammography, of stool-based tests, lung cancer screening, and so on and so forth. I do expect that to improve as the test continues to evolve. Yeah.
So Tina, you said something tantalizing before about the American Cancer Society guidelines. Can you talk more about that?
Sure. So, I mean, I think that is something that that we will need to evolve with the approach of these tests. So, just to backtrack a little bit about what we were just talking about, the screening test, there's a screening test and there's a diagnostic, right? So if you have a positive screening test, you're still not diagnosed with cancer. You still need to have some sort of diagnostic procedure. Eventually, maybe we will move these blood-based biomarkers into the diagnostic realm, but as of now, the way they're being developed is a screening, and then you still need to go in and have a colonoscopy or a lung biopsy or a bronchoscopy, in order to make the final diagnosis. So I think we do need to differentiate between screening and diagnosis. And that also comes into play when we're talking about the pathways to reimbursement. So there's different reimbursement strategies and financial recuperations for diagnostic tests than there are for screening tests. And Tom alluded to that with the Medicare reimbursement strategies. But okay, so getting back to the guidelines. So I think another thing that, an area that we need innovation is coming up with data sets to support making a guideline. There's a certain amount of data that you need for getting a test FDA approved and into the marketable realm, into the commercial realm. And then there's a separate level of evidence that you need for putting it into a guideline for screening. So where can we draw on those data sets? I mean, this is not my area of expertise with respect to the actual statistical evaluation of the data, but that belongs to my colleague, Bob Smith, in the intramural program at the American Cancer Society. But I do think that's something that should be up for discussion. I think we need innovation there. What level of evidence do we need to put it into a guideline? Do we need a 300,000 patient clinical trial that shows overall survival benefit over 20 years, or are there some other surrogate markers that we can use? Tom, maybe you can talk to us about that.
I could talk about that forever if you let me.
Go ahead.
Well, I think there's no question that we all want to reduce mortality from cancer by screening. So there's no disagreement about that. The debate in the field is really, how do we get there, and do we require prospective evidence of a mortality benefit before we begin implementing these tests? And that's where there's some disagreement in the field. I think, a requirement for mortality would mean a 15 to 20 year period before we really make a dent in the 600,000 deaths per year that we have in the United States. On the other hand, we don't want to be too cavalier and implement something that doesn't really turn out to be useful in the long run. So we're thinking hard about that. We're thinking about what level of, for example, decrease in stage 4 cancer or decrease in the clinical consequences of having advanced cancer, for example, the rate of disability or cancer-related symptomatology that we might be able to pick up within 3 to 5 years as a early indicator of benefit that, if it's robust enough, might be persuasive. We haven't settled on how to do that well, but we're pretty clear that if mortality is the only way to go, it'll be quite difficult to attract investment in this space or to really make a difference for the people that we know that need the help sooner than that.
And I would agree with you. The idea of having overall survival as the endpoint will take years and years. Many people would die. So we have to figure out something else like stage shift, as you know, of course, the thing we look at. And there are models that have been done, which are okay. And they seem to indicate that, yes, we may not have to do overall survival. And I agree with you. If we wait for overall survival for a very long time, then we won't get investment. So stage shift almost has to be a new endpoint or some sort of thing similar to that.
Yeah, and I would say, that's something we've talked about, the role of PMC, just as a sort of somebody who's, a clinician. I think that it would really be beneficial for a coalition like PMC to advocate the FDA or other organizations to change some of these endpoints. Because I think it also really prevents people from getting into the field, from companies getting in the field, because right now we need such long trials with such, really ambitious endpoints to be even able to offer the diagnostic test or get coverage for it.
And I think the American Cancer Society is very involved in that. There's a multi-cancer early detection consortium. So there are groups that are doing that work and thinking about it. Blood Pack is another. So that work is ongoing, but of course, PMC could meaningfully contribute as well.
And Tom, you are right. We do have to be very careful because there's micrometastases all over the place. And so you may look like you've done something in stage shift, but it's also possible that there's micrometastases that can come out. And that's why overall survival is still important. I don't have an answer. I want to just raise one other issue which we haven't talked about, which the previous panel did, which is disparities. We all deal with that. And in the case of early detection, we deal with it. Everyone thought that early detection would be that it would close disparities. The opposite. There's more disparities now in early detection than there was in 1970. And that's for a whole host of reasons. It has to do with get early detection. You get early detection. Let's give an example, breast cancer. White women and Black women both have the same incidence of breast cancer, and yet the mortality is 40% higher for African-American women. So what happened is that test actually increased the disparities. Prior to those tests, it was about the same mortality rate. What happened? A number of things have happened. It takes a long time, by the time an African-American person with all the children and issues that they have to take care of, jobs, and so on, to be able to get treatment quickly. That's just one area. Then the other thing is, in Washington, D.C., there's a study that showed that in wards 7 and 8, which are the poorest wards, in Washington, D.C., 50% of people in those two wards were diagnosed with cancer, were unable to follow through on treatment because treatment's really hard. You have to give up your job, you may have children at home, and so on and so forth. So we have an issue of disparity on early detection that people don't talk about, but it's a very serious issue in every single area of early detection, lung cancer, MCTs, and so on.
That's a really good point. Does anyone want to
I
address
mean,
that?
absolutely. This is something that we definitely need to keep in mind. As we increase our innovation, how are we going to keep a health equity lens on that? Happens in lung cancer screening, happens in cervical cancer screening, not just breast cancer screening. I mean, it happens in every possibility where there's a lack of access to healthcare. So, and that's in inner city people, but it's also in rural areas as well, where there's a lack of access to high quality CT scans and high quality readings of mammograms, for example. So I think, yeah, any test that we develop and get finally through the Medicare reimbursement system, however we do that, will be important to have a health equity lens on that.
Yeah, but it's not only a reimbursement, because Medicaid is in the sense of reimbursement. It's the problem that low-income people do not have the time. They're unable to be able to go through the whole process of what I call the labyrinth of cancer. That's really what the problem is, and that's what we work on heavily. That's my orientation to disparities. And what we find is that there are so many social determinants that determine that you can't really get treated properly. And the hospital differences. There are very good hospitals on York Avenue. I'm from New York, I'm from Brooklyn. First Avenue is filled with the greatest hospitals in the world. You go into Brooklyn, you go into Queens, that's not the case. We work with Jamaica Hospital. Jamaica Hospital, 50% capitation. There are only 8 hospitals in all of Brooklyn, all of Queens, which is 2.5 million people, 120 separate languages, 8 hospitals. So it's not this issue of parity and disparity is not only an issue of individuals, it's where the hospitals are also and the quality of the hospital.
Okay, so how does developing new screening tests address that, or does it not? I mean, how does developing new screening tests make a difference there?
Correct.
It does.
So,
are you saying it doesn't?
It does make a difference.
How does
but you've got to learn how to implement it. And I've done implementing experiments. We've used vans. We've tried all kinds of different things. And actually, Tom said something very important. He said that colon cancer was almost cut in half. And it was almost cut in half because the CDC spent $300 million or got $300 million in free advertising over a period of 15 to 20 years. They did 225 focus groups to tell people about screening. And that cut colon cancer down by between 40 and 50%. So mass media does work because people watch TV, there's social media and so on. But there's nothing like that that's been done with lung cancer. There's nothing and that's one of the most important ones for whatever reason. But to your point, it really worked in the case of colon cancer.
And I think it's also, diversifying the workforce and getting into the community. So one is general awareness, which is really important in the mass media, but the other is really community-based education and bringing the vans out and making sure that as you implement MSAT tests, that you're working with the primary care providers who are in those communities. And I think as we build our programs, we're really cognizant of that and getting the community engaged right from the very beginning as you're developing the programs to see what do they need, what are their barriers, and make sure that you're making the process, not cumbersome, as efficient and efficacious as it can be. Sorry, go ahead.
I've got a lot to say on this. Do we have time?
Few minutes, yeah.
Well, well, no, I mean, so there's so many dimensions to this issue. I'm not going to address all of them, but I just want to highlight 3 or 4. So with MSED and really any blood-based test, one of the critical steps in terms of where we are right now, which is test development, is that we begin by including everyone from the beginning. These tests are designed using human specimens and human cancer diagnoses to select the algorithms to detect the cancer. So they have to be done with everyone and for everyone from the beginning, from the first case control studies that lead to algorithm development, and then inclusion in the prospective studies so that A, we know that these tests do indeed work equally or similarly for all the groups, and B, everyone can have confidence in those tests when they become available. So those are steps we're thinking about right now. Broad reimbursement is crucial. I mean, there's not much to talk about with regard to equitable access until people can afford these things. And so it really begins with an FDA approval and then access to reimbursement. One of the things that we at Exact can bring to the table to this is the experience of Cologuard, which is reimbursed without a copay for 96% of Americans. And that's enabled us to really engage with federally qualified health centers, with all kinds of communities to try to be to address this issue as much as we possibly can. One thing that we've also been able to do is offer customer support in 240 languages, for example. So there's things like that we're learning and working on, and I can't represent them all accurately, but I think there's a lot you can do, but we do have to be thinking about that from day one, not after we get the test to market.
All right, to change the subject a little, one thing I wonder about with early detection is what are you detecting? How do you know that the cancer you're detecting is going to grow into something that's a problem? And how great is the risk that you're going to pick up all these signals that aren't really going to make a difference in people's lives? I don't know, Tina, can you talk about that?
Well, I would say, going back to what I was saying before, there's a difference between a screening test and a diagnostic test. So the screening test would then trigger a diagnostic procedure. So, for example, if you had a blood test that detected colon cancer, you would then proceed, or that suggested that you had colon cancer with a 60 to 80% specificity. You would then proceed to a diagnostic procedure, which would then be a colonoscopy. You would take a biopsy, you would see how much invasion of the tissue there was. You would get a stage of cancer and a diagnosis and a stage and maybe even a subtype of a cancer. So that's a long answer to say that I don't think that the screening test itself is going to necessarily give you the full picture. The full picture. So you need to follow up on it.
But that sounds very costly. What do we expect this to do to the cost of healthcare if that many more people are undergoing these further evaluations that will, in many cases, turn up nothing?
So it's indolent cancers, is really what you're talking about. And so it's one of the most difficult problems that we all have is what is an indolent cancer? And I'm radical at this point. I don't think any cancers are really indolent because if you look at ovarian cancer, it may start out as indolent, but 2 years later, it can get aggressive. Now, that's not necessarily as true with prostate cancer and other cancers, but the problem is it may look like it's indolent, but it may come back. So I'm somebody who says, you know what, if it were me and I had an indolent cancer, someone said an indolent cancer, I would take it out because I can't guarantee that it's not going to 2 years later get worse. But it is, despite my radicalism about it, it is a huge problem.
Yeah, but so I'd take a slightly different view with all due
I knew you
respect.
would.
And, I was a prostate oncologist before I took this job, and in prostate cancer, this concept of overdiagnosis and overtreatment was very real. But I do think it's important for us to acknowledge that the field of prostate cancer, urology, oncology responded to that. It took a while, and we didn't learn overnight, but approaches with active surveillance that decoupled diagnosis from treatment, and even nowadays approaches to reconsider whether low-grade prostate cancer is something that we should really be diagnosing in the first place. So when we encounter situations like this, we need to learn and improve. And I think that captures potentially the benefit of prostate cancer screening while reducing the harms. The blood test that we're working on now, at least in the multi-cancer space, time will tell. We need the evidence, but by virtue of the nature of the types of biomarkers, what we're seeing is lower than hoped for stage 1 sensitivity, good stage 2 and 3 sensitivity, really great stage 4 sensitivity, but that's not what we're trying to do. But when you have relatively low stage 1 sensitivity, it seems unlikely that we'll be picking up really small indolent cancers. They just don't spill a lot of DNA into the bloodstream. So time will tell, but I'm less worried about that problem for MSEDs than I might be for a PSA test.
Yeah, I agree completely. MSEDs don't essentially pick up. Not enough.
Okay, every woman who's had a mammogram and got that phone call, we saw something, we need to come back and look more deeply, and then found out they were fine, will tell you that there is a cost to screening, right? A really big emotional cost, as well as the cost of the treatments. And how does that figure into the future of these tests? What thoughts do you have about exposing more people to the thought, oh, you might have cancer? What does that do?
Well,
Wait,
Should I go? So again, that's a reason why it takes so long for the American Cancer Society to put it into a guideline. You have to take all those things into account. What is the cost to society? What is the cost to the patient? What is the financial cost? All these things need to be taken into account in order to say that this is something that should be done. Lots of studies need to be done. This could take, that's why it takes so many years to be able to generate the evidence to say that it's worthwhile doing. But these are all excellent questions and things that we need to keep in mind. But that being said, I mean, if you have a thing on, a spot on the mammogram and it is cancer, then you want to know that. It's not that you don't want to know it, right? It's better to go get it done and not have it taken out because it's nothing.
I do think, so what from a statistical standpoint, we're talking about test specificity, right? So which directly links to the risk of false positives. So just to put a little bit of math around that, our current generation of single cancer screening tests have, between a 5 and a 15% false positive rate, let's say. The MSED tests are being designed to have about a 1% false positive rate. So hopefully it will not be an enormous number of people. It's certainly a much lower risk than it is with the single cancer tests of today.
So wait, okay. So are you saying that 98% of the people who get this test and they get the answer that you have cancer will definitely have cancer?
No, what I'm saying is that 98 to 99% of the people who take the test get a normal result. And so they do not get told that they might have cancer.
Right, but among the people who are told they have cancer, 98% will really have cancer?
No, that's the, we don't know yet, but that number is likely to be about 1 in 3 have cancer and 2 out of 3 do not. That's, for stool-based tests, that's like 1 in 20 have cancer and 19 out of 20 don't. So those are kind of the numbers, but if you're in a screening program, then the number of false positives is what starts this conversation. And it's about 1 per 100 for MSET. It's about 10 per 100 for standard screening tests.
And the other piece that, just to add one thing is that it's really important that for the reimbursement, that it's not just about the test. Because the follow-up and the evaluation of the test have to be covered. And then if you, let's say you're found to have a positive MSET test, but nothing is found on the PET scan and the colonoscopy and all that stuff, and you want to do it again, I think it has to be bundled together, because in addition to the emotional, duress, the financial toxicity potential is huge too. And, we've seen this in germline genetic testing. We were trying to get pancreatic cancer patients in underserved communities to do genetic testing, which is guideline recommended. And across the state, there's so much variability in coverage for pancreatic cancer screening if you're found to be a germline mutation carrier, that nobody would do the test. So you really have to think about the whole picture, not just how you get into the system for screening.
So you asked about breast cancer, and I think we talked about mammography. So there's something called recall rate, which means what you were talking about, you've got to come back. So in the United States, our recall rate is about 20%. In Europe, it's between 8 and 10%, which means we recall twice as often. And the reason for that is probably a very simple reason. In order to do mammography reading in this country, you have to do approximately 480 mammograms a year. In UK, it's 5,000, meaning that much better reading is done in Europe. And that reduces recall rates in half. And so that's what I think you're talking about. The anxiety, and I know it, there's studies on it, the anxiety of a woman having to come back for a recall is very scary. We've got to reduce it as much as possible, which really means we need higher standards with respect to reading mammograms. We've got to get to rates where you need to do 3,000 or 4,000 a year in order to be allowed to do mammography reading. So I think that's what you were kind of referring to.
It was, but I'm also just thinking about what the future holds with these tests becoming more available. And that sort of leads to my next question. And by the way, if anybody has questions, come up to the mic, because I think we'll have time for questions, right? Yeah, okay, but all right. So, but the question I'd like to ask all of you is, where do you think we'll be in, say, 5 years? What's the future? How fast is this moving, and what's it going to look like? Get out your crystal balls.
We'll have another president, maybe.
Tina.
I was going to boot it to Tom. No, I mean, I think we will still be generating evidence in 5 years. I mean, I think we'll definitely be closer, and there'll be more tests in the pipeline, I should say, in the pipeline. But I can't see in 5 years that everybody's going to all of a sudden be getting an EBSET. So I think it's going to take longer than that to get it into practice.
Yeah, I don't disagree. I'd love to see us move faster, but when you really think about all the steps that are involved from where we are today to widespread adoption, I think we'll have a lot more data to talk about. We'll have answers to some of the burning questions of today, but it'll likely take a bit longer to get across the finish line.
10 years? I probably should ask 10 years.
Well, I do think, so the American Cancer Society Cancer Action Network is working on the pathway to reimbursement. So I do think we would have at least a pathway to reimbursement 5 years from now.
Okay.
Yeah, I think you're being a little bit pessimistic. I don't think we'll have all the answers, but we'll have many more answers. And I think there'll probably be more people getting tested than there are today. So I think, some of these, there's clearly going to be disparities. There may not be universal reimbursement, but there is in the clinical community, for the patient community, real interest in the test. I think from the scientific perspective, policy, reimbursement, we have to be really careful. But I think we're going to have a lot more data in 5 years that will help us guide the decision. At least know, is it going to go forward, or is it, is it moving? Is it a done deal and this was a wrong path to go down?
So I think that we're going to know a lot more, as I said. I think on the other hand, when you look at cervical cancer, it took 60 years from 1928 when Dr. Papanopoulos came up with the cervical cancer pap test. It wasn't until 1988 that it was really perfected and used. Lung cancer, 1990, Claudia Henschke came up with the idea of low-dose CT scan. And yet only 5% of people actually use it. So I'm pretty pessimistic about the implementation. I'm not pessimistic about what you said, which is discovery.
Okay.
So I've got maybe a comment and maybe a request for comment. And Tom, this is a little bit directed towards you. So in small and large molecules, health economics outcomes research is really the norm now. And it seems to be that the payer is missing in this conversation, either missing on stage or missing as part of the debate in terms of being able to be part of the process. There's a clear intuitive savings potential here, not just with diagnostics, but potentially with screening as well. Having worked at United Health Group for about 6 years in my career, ran the formulary there, I actually got a Cologuard test for free, sent to my home, and I worked for a payer. That's how bought in they were. So I just wanted to maybe get a comment on how you were clearly successful and others need to follow. Is it a matter of not bringing that payer in early on versus going after pharma as the client? Because there's a lot of vertical integration in the small and large molecule payers today. Seems a logical step that would be where diagnostics and screening would go to.
Yeah, I couldn't agree more. And yeah, I think there are many, or there are other forums where payers are engaged. For example, in the MSED consortium, where they're part of the discussion internally to Exact, we have a very strong and talented healthcare economics and outcomes research group, which is well known for its colorectal cancer work and has begun to present its MSED work. And it'll certainly look at things like cost of cancer care at various stages and work to translate a potential stage shift into healthcare economic outcome predictions. And I do think that the healthcare economic case is more complex than just the incremental cost of the testing. It's also the downstream evaluation costs and the savings, hopefully, from earlier diagnosis and earlier treatment. So we will be looking at that.
I thank you, Felice, and thank you to all the panelists for a great discussion about what I think is one of the most important areas in personalized medicine that's developing. So thank you. I really appreciate your comments and all of your statements about how difficult this will be to implement and provide access to these screenings. And I think you really put a good picture together on what those challenges are. But one of the arguments or one of the things that have been floated, ideas that have been floated, is to bring access to these through a risk-based strategy to at least make sure that those highest risk patients can get screening, either multi-cancer or individual screening, so that we can, at least bring this forward from that strategy instead of trying to bring it all together at once and to have to deal with all those implementation strategies. So what are your, especially in light of all of the 5-year projections that you guys gave, what is your thoughts on bringing it together to bringing this forward to a high risk population? And what would be the strategy for determining who should get access to these tests on a risk-based strategy first?
You're talking about high risk people should be screened earlier. Is that what you're saying?
Exactly. So that we can make sure that we provide that
I
reimbursement
think
to high risk patients.
I'm using funny 2 words. That's the holy grail, I think, in a lot of ways, and I don't need that kind of grail. I think every doctor I speak to and all the people I deal with in early detection of cancer, what you're talking about is one of the most important things. How do you detect, or find rather, those people who have higher likelihoods of getting cancer, and therefore they should be tested? There's a lot of work being done by a lot of different scientists, doctors, trying to figure that out. And so there's really good science work that's being looked at on that question. But it is really important, to your point. We shouldn't have to screen the entire United States. We should be able to know who is more likely to get cancer and really focus on that and give them more testing and so on. So I don't have a good answer, really, other than That's a really important issue that is being looked at by many, many scientists because for all the reasons you just said.
Go ahead.
Okay, so yeah, so I completely agree with you. I think risk-based screening is certainly a logical strategy. That brings up another point as maybe this could be a developmental strategy is to kind of validate the test in those high-risk patients that are already going for or that are already eligible for known screenings. And that's going to, obviously limit it to the 4 cancers that we have screening for: lung cancer, colon cancer, cervical cancer, and breast cancer. But that could be a start to sort of validate this and then take it forward. We don't really have clear, as you mentioned, clear risk stratification for the other cancers because there hasn't been screening tests for them to date. I mean, we have obviously the mutation carriers for various cancers, but we have a long way to go.
Yeah, we tried to get sort of a force behind this for a long time to think about the really high-risk groups, the mutation carriers, and ultimately ended up self-funding it. So we're conducting a trial right now using MSED testing in 1,000 patients. Half of them will be mutation carriers, so either BRCA or Lynch or any of their high-risk cancer susceptibility genes, and the other 500 will be people with a family history. So I think that is really one of the strategies I think that's worth exploring, is whether these tests will have better value in high-risk populations rather than, the bar will be different and the prevalence of cancers might be very different.
Another question?
Yeah, I'm Alan Sachs, Thermo Fisher Scientific. I've seen a lot of the liquid biopsy companies sort of reverting to MRD as sort of an intermediate step because MSED is going to be a longer term. How do you see the data relationship between MRD studies and supporting MSED work, or is there any?
The what relationship?
The relationship between the minimal residual disease work and all the evidence needed to get reimbursement for that, and then later the MSED work, which may use some of the same markers. I'm wondering, are they connected in your world or are they really separate?
I can't speak to all aspects of your question, but I mean, I think the evidentiary standards are different for those 2 indications, and the test performance characteristics are also different. So, I mean, there's some overlap between these 2 approaches, but when you look at MRD, you're looking at initially some very specific clinical decision points that can be influenced by an MRD test one way or another, adjuvant therapy versus not, or early intervention versus not. The performance of modern MRD tests in terms of sensitivity is a little different than the performance of MSED tests of this generation. So it's not exactly interchangeable in my mind. There's some overlap in technologies and potentially some overlap in indications for use. MRD negative patients are at risk of other cancers. Could they also take advantage of an MSED? Perhaps, but I don't see those as interchangeable.
I think, can we take one more question? Yeah, we're cutting into your networking break, but this is all really good. So go ahead.
Hi, Josh Hoffman from Grow. Maybe just to comment on, thanks for the great panel. This discussion you were just having about risk. And, we do know that age is by far the most important risk factor for cancer and overwhelms almost every other risk. In fact, we know a lot about the relative risks of obesity or alcohol use or viral infection, but from an absolute risk perspective, one age of additional age year over the age of 50 is more important than many of those other relative risks for cancer. So we in fact do know, who is at high risk for cancer. If you start adding the hereditary cancer syndromes with age or survivorship with age, we do know that is high risk. So I would encourage people to think about age as probably the single most important thing we can do to identify those who are at risk for cancer. Thank you.
So, and I would amend what I would say. I agree with you that age is critical. And that's why the reimbursement for almost all these cancers, for the 5 tests that there are, start at age 40 or 50, because those people are more likely to get cancer. So in some sense, the insurance follows your point that obviously age is a definite risk, obesity is a risk, and so on. So it is done by reimbursement. Over age 50, get reimbursed, or over age 40 or 45, depending what test it is.
I will say one little comment about risk-based applications is it's remarkable to me how simplicity of implementation drives adoption. So, I mean, there's lots of reasons why lung cancer screening is done so poorly, but the table of requirements to whether you qualify or not, you think that's simple, but it's a big barrier. So yes, I think starting in defined populations makes a lot of scientific sense and clinical sense. But in the long run, if you create very complex decision trees for who qualifies and who doesn't for screening, that's going to substantially depress adoption.
And I agree with you, because I do lung cancer screening with vans and so on. You have to go get a pre-authorization from the insurance company. You don't have to do that for breast cancer. That's a real hassle, because about 20% of the time, the insurance company doesn't believe that you were a smoker. So you've got to go fight that out. And then you've got to do smoking cessation with the individual and teach them about it and talk about it. And then a shared decision, 3 different things. So very often you have to have the person come back twice. And we're working with Jamaica Hospital. In the case of Jamaica Hospital, they make 6 calls to get 1 patient to come in. The patient comes in, they then have to check all those 3 things I mentioned, and they have to come back another time. So your point is that there are a lot of impediments which are just shouldn't be there, that are there, particularly on cancer.
Right, I think we've run over, but since we're already over, if there's any vital point we haven't covered, speak up. Otherwise, thank you so much. This was really a great conversation.
So we are at our networking break. We'll be back here at 3:45, and please do visit the upper exhibit hall. It is literally true that this conference is impossible without them, so please do go upstairs and say hi. Thank you.
Hello, everybody. Welcome back to the afternoon session for the home stretch. It's really a great privilege to introduce my friend and colleague, Paul Billings, who's been doing personalized medicine for longer than I've been at PMC, has a long history in the space, which maybe he'll tell us about. But when he told me that he thought that this year's conference should do a session on pediatrics and personalized medicine, we said, Paul, that's a great idea, but only if you agree to moderate it. So with that, I'd like to turn it over to Paul.
So thank you to all my friends at the PMC for allowing me to participate again for, I don't know how many years. And I too want to add my congratulations to Ed for all that he has built over this 20 years or so, and for a happy and healthy transition into whatever you decide to do in the future. So I'm Paul Billings. I've been a long-term member of the PMC. I'm a clinical geneticist and internist by a professional DIN, and have had the pleasure of working with some or many of you in the room in various activities. That's how I'll identify myself for now, and now I'll let my colleagues here on the panel identify yourself. Go right ahead.
Hi, I'm Mira Irons. I'm the Associate Chief of Genetics and Genomics right up the street at Boston Children's Hospital. Trained in pediatrics and then in medical genetics. Just for some context, which may become a little important in some of what I say. I completed my training in genetics in 1986, which I define as the pre-clinically molecular era of genetics. But it actually provides an interesting perspective into how far we've come since that time. I recently came back to Children's. I was also the Chief Health and Science Officer at the American Medical Association for a few years, and the Senior Vice President of Academic Affairs at the American Board of Medical Specialties. So worked with the specialty society, the specialty boards on certification and continuing certification.
Hello, I'm Lillian Garbin. I'm from Nicholas Children's Hospital. I'm currently the Director of Operations for several of our programs out of Center of Precision Medicine. My training is I'm an epidemiologist and a biomedical engineer. And prior to my time at Nicholas, I was the lead epidemiologist for our adult healthcare facility down in Miami, Florida. Thank you so much for having us today.
My name is Julia Vitarello. Thank you all for having me. I am first and foremost Mila's mom. My daughter, Mila, I was asked to explain a little bit about the context of why I'm here. My daughter Mila was the first person in the world to receive a medicine designed for one person called Milicin. My life changed overnight when my daughter was diagnosed with a fatal, 100% fatal, very rare neurodegenerative disease. And my outgoing little girl that had been skiing and running and biking felt like overnight lost her vision, her ability to talk, to walk, to eat, and I was told she was going to die. And in my fight to save her life, together with Dr. Timothy Yu, who will be here coming tomorrow at Boston Children's Hospital as well, over the course of a year, became one of the fastest things to go through the FDA at the time, and started a treatment for my daughter. And the catch was that she was the only person in the world who shared that specific mutation that her antisense oligonucleotide targeted. And of course, as you can imagine, many people said to Dr. Yu and us, this is a crazy notion. How can you have a novel medicine for one person? But we did it, and it stopped my daughter's disease in the first year, despite her incredibly, very rapid, horrible progression leading up to that. But it was not in time for my daughter, and she died at age 10, 3 years ago. And the reason that I'm here, and why I do what I do every day, is because it wasn't in time for my daughter, but it showed what's possible. It showed that we now have the technology to find the underlying genetic cause, often a single mutation, in the case of my daughter, and design a medicine for it, even if it's unique to one person or just a few. So to me, that is individualized medicines, and I'm really grateful for being part of this panel. Thanks.
All right, well, I'm going to make a couple of preliminary remarks and then ask each panel member a couple of questions about how pediatric applications of precision medicine might be the same or differ from what we're seeing roll out in the adult world. Pediatric medicine is different, right? Some of your patients can't speak and haven't learned how to speak yet because they're too young. And almost all of them have short clinical histories because they've had short histories, period. Kids have been neglected in many types of clinical trials historically. And some of those clinical trials have sadly been unethical and inappropriate in some ways. Kids, the pediatric market is smaller than the global adult market. And some things are promising and known about the pediatric presentation or pediatric medicine. One important one is that, and this has been known for some time, that around 30% of the early hospitalizations in the pediatric population are for monogenic genetic disorders. Those are increasingly identifiable, if not all identifiable now. And we just had some recent data published that something like 30 or 40% of the NICU, the very most expensive part of most every hospital, about 30% of the NICU admissions are also for monogenic genetic disorders, which are identifiable and either treatable directly or treatable outside of the ICU, or not treatable at all. So from my point of view, adult medicine historically has been driven by two things. One, symptoms, which often requires you to describe them to your physician, and an expert system of providers who use differential diagnosis to come to a diagnostic in a patient. Now, that system, while obviously has yielded some very good medical care, has also produced medical odysseys that could be years in their length, misdiagnosis, ineffective treatments, and we all know about that, and we actually have heard about that a little bit already here today. Poor outcomes in some large classes of diseases, and a very expensive hospital system, which accommodates that old system of presentation. It also, by the way, has generated a clinical trial system which highly relies on prospective randomized clinical trials. Now, but with Ed's work and the work of many of the people in this room, we are seeing a change, a evolution of a new system of personalized medicine. It relies a little bit more than the previous system did on asymptomatic or pre-symptomatic risk identification. And we're seeing preventive interventions to deal with that risk identification effectively. We heard a panel just a little while ago about early detection. That's increasingly an important part of the new adult medicine, precision medicine paradigm. We're seeing the application of computers rather than experts, computers playing an important and assistant role in the provision of information and diagnosis. So what's likely to be the result of that evolution of precision adult medicine? Well, faster diagnoses, hopefully more accurate diagnoses, more targeted treatments, maybe a less costly healthcare system. And I think also some more reliance on N of 1 trials or certainly smaller, more targeted trials to get to approvals and cross-regulatory hurdles. So how is that new model that's evolving, and not all of it, it's not evolving uniformly, but is evolving, how might it be implemented in the pediatric setting? Well, one thing that's good news about it all is that screening happens in pediatrics. You can do a whole genome sequence on a fetus. And obviously, non-invasive prenatal testing, which gives you information about the fetus and the mother, is widely implemented. We have a system of newborn screening in all parts of the United States and many parts of the world. And that is moving rapidly to whole genome sequencing as a model for how that newborn screening will take place. We're obviously have lots of rare and genetic disorders in the pediatric population. And I think that the focus on them and development on them, even though they're small markets, will continue. And we have new kinds of clinical trials that will hopefully be well established in the pediatric population, and which will take into account that kids have developmental systems. Many of their systems are not fully developed until they're long after they're born. And many of them have neuropsychiatric development to do when they become sick and become part of the medical care system. And that's not so important, or is usually not taken into account in adult medicine trials. So those are sort of unique challenges in my mind and unique aspects of the evolution of personalized medicine in the pediatric setting. I'm sure people in the audience can think of many other unique things about the pediatric population. But now let's turn to our panel. And Mira, why don't I start with you? What does personalized medicine, or if you want to call it precision medicine or individualized medicine, what does it mean to you? And how do you think it's different from classic medicine, what you were trained with in the 70s and 80s? And then what's unique about it in its application to pediatric patients?
Thanks, Paul. I think when I go back to when I started as a pediatric geneticist, we didn't really have very good diagnostic testing. And as Paul said, 30–40% of children in hospitals had some genetic condition. Many of those kids in the first year had multiple physical abnormalities. There was intrauterine growth retardation. As they got older, we started to see children with developmental delay, intellectual disability, autism. And many of those patients were my patients. They came to genetics because they were looking for a diagnosis. They didn't fit They didn't fall into any specific category. So when I think of personalized medicine, you know, we were all waiting for this day. We would take care of these kids that came with multi-system conditions that didn't fall into any category. We made clinical diagnoses at the time, but we would always say, you know, at some point in time, our diagnostic testing is going to get better. It's going to get good enough that we'll be able to identify the gene that causes your problem. And once you have that diagnosis, then the next step is therapy. And we're there now. I think, you know, it's safe to say that we're almost there. I think there's a lot of promise, but as Paul said, there are challenges. So when I think about this, I divide these things into two categories. One is diagnosis. You know, the healthcare system really revolves around patients coming with symptoms to see a healthcare provider, right? So they have something and you go down that path. And those kids I would divide into three groups. One of those groups is children with known conditions that you can diagnose by other means: cystic fibrosis, sickle cell disease, Duchenne muscular dystrophy. And I think we've come the farthest with personalized or precision medicine in those kids, because identifying the genetic changes that they have has led to gene therapy in some of those patients. So that's one distinct group. The other group is the group of kids with what we call undiagnosed disease, or used to be called rare disease. And I think we finally made a difference in those kids because, to be honest, because of exome and genome testing. We can now make a diagnosis in about 30 to 40% of those kids, which is huge in my world. And it's the hope then is that genetics as a specialty will move from diagnosis to therapy, which you'll hear about, you know, from Mila's mother. But there's also a third group there. And that third group are the 50% that are not diagnosed. And those are the kids that are still looking for a diagnosis, or on that diagnostic odyssey. So we're not there yet. That's the diagnostic piece. The other piece, and we've talked about it in some of the earlier panels, is prevention. As Paul said, newborn screening, which started in the 60s for phenylketonuria, is really the oldest, I think, example of personalized medicine. And it works in pediatrics. They're state-based programs. It's getting bigger and bigger, and we know how to take care of those kids. The challenge is going to be with newborn genome screening. There are 3 or 4 different, and there are probably more, but there's the Guardian study, Baby Seek here in Boston, David Bick's work in the UK, looking at newborn genome screening, which I think is where a lot of us feel we need to go. But what has to come from that is we need to know what we're going to do when those children are identified. We need practice guidelines. We need somewhere for those kids to land. And that's, I think, our challenge. But I think that's the greatest way we can impact child health, but there's a lot to do to lay the groundwork for that.
Do you think that telling parents of a newborn that they have a risk, some of that risk may manifest itself during the pediatric years, but let's say it's a risk that may have its main impact in adult years, is that going to create a whole new conundrum, a new kind of anxiety that parents are going to have to deal with?
Well, I think that the research studies, Guardian study, the Baby Seek study, are research studies to begin with to actually answer that question. I can tell you that, you know, we used to have all these rules. And one of the rules was that we would not test a child for any condition that presented after 18 years of age if they were at risk for it, because then they could decide when they're 18 if they wanted to be tested for it. I think newborn genome screening could change that paradigm. We've kind of done the test already, because when you send an exome on a child with, say, seizures or or autism. There is, the parents can opt into testing for secondary findings. And I think it was actually addressed earlier, the ACMG 89 different conditions. And those are conditions that can manifest during childhood that parents can either opt in or opt out of. And I can tell you anecdotally that most of the people that my group talks to, they opt in for the findings. And the promise there is that many of these conditions are autosomal dominant. So if you actually diagnose a child as having a BRCA1 mutation as part of secondary findings, you actually can then diagnose one of the parents and other family members that may not have known that they were at risk. So I think we're already starting to gather some information on that, but there are not significant, but there are groups of people that just opt out. So,
yeah. And I think that's a very important point to say that testing someone in a family begins to unravel the mystery of that family's genetics. And I think that's, and it's also true that for clinical trials, whether those clinical trials are going to take place during pediatric years or in adult years, there can be more people who are genotyped coming into the clinical trial availability pool, and that will allow some precision in those clinical trials.
I think the one thing I would say, though, is that these conversations take time. You know, to consent someone for an exome and to talk about secondary findings and consent for that, you know, takes at least 15 to 20 minutes. And just think about how that impacts the healthcare system. You know, internists, I'm told, have these 8-minute blocks of time that they see patients. You can't put this consenting or this information into that. So I think that that's another challenge, is how do we educate people to explain these things? How do we provide the educational material? How do we do it in different languages? How do we talk to families about genes when that's a foreign subject to people? So there's a lot of education we still have to do, but I do think we're at a pivotal time. I think we can really positively impact child health. But we have to do it thoughtfully and we have to do it ethically.
Right. So Lillian, what do you think about what's happening in pediatric medicine and what you see, and what are the challenges to apply precision and what's happening in the precision or individualized world in the pediatric setting?
So I think we've said it a couple times today that it's not one size fits all. We originally thought that was what the basis for medicine was, but we've moved away from that. We're now moving towards this precision medicine, and we see it in our clinic on a daily basis, especially in our oncology trial. One of the significant challenges that we have in a pediatric population that we don't have in an adult population is a child is constantly growing. They're developing. A child at the age of 2 is not the same child at the age of 6, at the age of 12, 16. An adult, although you have challenges, they're no longer developing, they're no longer growing, they're fully developed as a human. So we have to constantly be tailoring our approach again to go back for this same child just to be able to target their therapy. Going back to the oncology trial that we have, a case comes to mind of a child that has had 4 relapses. We've done precision testing each time, drug sensitivity testing to see what medication would their tumor respond best to. Each time we've had a completely different result because this child has relapses within 2-3 years at a time. And now as they've aged, their tumor has also changed. So they're no longer responding to what we originally had prescribed and we had seen successful in their treatment. So I think that's one of the significant challenges, but also to come from an angle of what does precision medicine mean to me and to my staff. Stripping back from the science and stripping back from the testing and all of our medical knowledge. I had a patient's mom approach me that when we had explained what these tests could mean and what it looked like in comparison to all the testing that their child had gone on this diagnostic odyssey, her first word to me was, "There's hope." And I think that's something that we as my team, we keep that at the forefront for us. We can speak science all day long to our patients and to our families, but what they see at the end of the day is hope and an opportunity for their child that has been on a very long journey without any answers to potentially have a better outcome from their disease, to have a higher chance at a longer life than what they originally had. And I think that's really what precision medicine gives us. It gives us hope, and it gives us new answers and a new direction that hopefully we'll be able to take it to the next level.
Do you have to temper that at some level? Do you have to caution because of, we've already illustrated that there aren't treatments for all the things that can be detected now. And we don't know that. We don't have long-term outcome studies for lots of things. So do you have to be a little cautious about that, or do you let that hope go wild?
So I think it's finding a good balance between the two. Remaining hopeful keeps us curious, and it keeps us in a spot where we're constantly pursuing diagnostics for our patients, treatments for them. But we also want to keep that reality for them that if it is a novel disease, if it's something that we haven't seen before, we're going to try our best. We're going to give them every possibility, every tool that we have, but there might not be that hopeful outcome at the end of the day. But we also like to keep in mind that if for that patient, we're not able to achieve the outcome that we wanted, hopefully that sets a setting stone for the next patient. And we're able to use that data, what we learned, those obstacles and those achievements to pave the path for the next patient that hopefully is able to have a better outcome.
Well, that was my comment about N-of-1 trials in the pediatric settings. Hopefully we'll be able to categorize more of those sort of individual experiences in databases which are shared and well curated, and be able to move some of these fields along a little more quickly because of that, or know that things don't work more quickly.
So when Mila was diagnosed with, what she was diagnosed with was called Batten CLN7. CLN7 is a subtype of Batten, a specific gene called the MFSD8 gene. And when she was diagnosed, but she was missing a mutation. It's autosomal recessive, and I was told by 10 doctors in white coats that she had this rare form of a disease where there are 25 known patients around the world. And there was 2 people in the world that knew anything about it, which was basically nothing. And there was no hope for her. So to your point of hope goes a long way. And what happened after that was that I was told that no lab could find this missing mutation. I did my homework and started, genetics 101 8 years ago, and sort of started understanding pretty quickly that whole genome sequencing at the time, which was the end of 2016, was very, very difficult to access, still is in many ways. And at the time it was $25,000, and it was a 5-month wait. And somehow I was going to raise the $25,000, but I couldn't wait 5 months. The reason being that I wanted to try to save my daughter's life, and I needed to know what gene it was. And when I went and looked at the databases, UCL databases on Batten disease in London, I didn't know anything about this. I just started noticing that there might be, 60 patients in there over many, many years, many of whom had passed away. But of those, some of them were missing mutations, and some of them had a third mutation in a different Batten-causing gene. So who was I? I just thought, well, maybe Mila has a different type of Batten disease. And if I'm going to give her one chance through, at the time, a gene replacement therapy, the only thing that I could do because we didn't know anything about the gene. How was I going to do that if I wasn't totally 100% sure that my daughter had Batten CLN7? What if she had a different form of Batten disease and one mutation in this CLN7 gene? And I also had a little son, and Mila had been completely typical. She had been skiing, rock climbing, when she was 2 years old, hiking for 4 hours on end. She was extremely verbal. She had 0 red flags the first 4 years of her life. And there was my little son, who was 2 at the time. And I needed to be able to test him. And without that other mutation, I couldn't test him, which meant that every single night I went to bed thinking that I was gonna have the one thing I wanted in my life is to be a mom, and to have motherhood just ripped from me for both my children was something I can never find words to describe. And so I was on a mission to find this missing mutation. And that resulted in me going, of all places, on Facebook and the desperate plea, can someone help me get into this lab at Harvard, which I had identified as one whole genome sequencing lab. And that then brought me to a different place in Harvard, or Boston Children's, where I met Dr. Timothy Yu. And that turned into the race that I just mentioned to you over the course of a year, which was breakneck speed with, I don't even know how many hundreds of people around the world helping to make this happen for Mila and for medicine as a whole. And that all started with the genetic testing. That all started with a missing mutation, which Dr. Yu and his team started to screen day and night, weekends for, I don't know, 1 or 2 months until they found this extremely rare retrotransposon. Don't anyone think in here that I fully understand what a retrotransposon is, but I know that's what it is, and I have a basic idea of it. But it was an insertion, a large insertion between exon 6 and 7, and that's what not only helped diagnose Mila and confirm her diagnosis, but it was the same information that was needed to then design her medicine. So I think this is something that's really important. We're thinking, especially around diagnostics, is like we are using whole genome sequencing to do the first thing, which is diagnose. But that information is now increasingly be able to be used to also design medicines. So if one brings up the topic of newborn screening, there was no treatment for Batten CLN7. So one could say, why would you want to diagnose Mila at birth? I don't know if I'd want to in 2010 when she was born, because I would have lost, to your point, all those years I had with her where I didn't think she was going to die. But now my answer would change, because that information then led to the creation, the design of a new treatment. And that is going to be the future. So when I look at what individualized medicines is, I look at hundreds or thousands of these rare diseases, which disproportionately affect children. There's 200 million children, according to Global Genes, that have a rare disease. If you do their math, which kind of who cares if it's exactly right, a third of those will die before the age of 5. That's 60 million. Does it matter if 60, 40, 20? It doesn't really matter to me. It's tens of millions or millions of children that are dying. And in these, they almost all have rare genetic diseases. They rarely have the more common ones that are commercially viable. So what really matters is that if something is not commercially viable in the current system, it means there's only 2 options. Either you're commercially viable and a company is going to make a treatment, or you have a crazy mom working with an awesome, physician to raise millions of dollars and have a 1,000-page IND with hundreds of people around the country and spending millions of dollars for one dying child. So this individualized medicines approach is particularly critical for children because they're disproportionately affected by rare diseases, which are not commercially viable. So there's zero hope for them, pretty much. And we know that treating early matters. Treating early is how we see, the best outcomes. And so what better place to treat than, ideally finding a child at birth or in utero and treating them immediately, so that the ultimate goal is that they and their families never experience life with disease. So if I saw Mila, I witnessed her at a very progressed state do incredibly well in her first year with her antisense oligonucleotide. She started laughing and smiling more, which was the most important thing to me. Her 30 seizures a day disappeared most days, and maybe a tiny little flinch once in a while. She could sit up straight. She could eat by mouth again. It wasn't perfect. It wasn't beautiful, but it was drastically different for us. And that was at, 7 years old, in a disease where kids die by like 9, 10 years old. Imagine if she had received that at, 2 months before, when she had no seizures, 2 years before at birth, ideally, right? Is we could have lived a life with that same drug that's out there right now, sitting on a shelf at Boston Children's Hospital. That next child that's born, maybe, gets, diagnosed at birth and receives Millicin. And they may never know what life with Batten disease is like. That's what we need. And individualized medicines, why does that play a role in pediatrics? Because there is no other bridge of access right now for nearly all of the millions or tens of millions of children that are living what I lived through, which I'm not sure how any parent, truly, I'm the most positive person in the world, and I don't know how anyone lives without their child. It is unbearable every single day. And there's parents that reach out to me every day, because Mila's story's been all over the world, and we have to change the system to accommodate this new paradigm. Otherwise, it's not that we can't. We used to not be able to, but now we can. We have technology to find these kids, and we have more and more tools in the toolbox to be able to treat them. ASOs are just one of them. There's many gene editing techniques coming from right here in the city that are going to be able to treat even more, but there is no pathway to access for an individualized medicine. There are few drugs for large populations, not many drugs for small populations, and that paradigm is what needs to shift. Otherwise, we're going to be here for sure in 10 years.
Where would you start? I mean, that's an
Conversation.
amazing story. So it sounds like everyone in the room agrees with you that we would like to see that shift, more therapeutic options as early as possible in people particularly with rare diseases in the pediatric population. Where would you start with changing the system?
It's a really good question. I mean, obviously, sometimes there are days it feels like the water is sort of spewing from all the different parts of the pipe, and I don't even know where to start. But I think it does start with regulatory, is a very obvious one. This is not to throw FDA or anyone under the bus. I think that the regulatory system was not designed for this paradigm, right? It was designed for, safety reassurance sort of like equals FDA, and every step of the way for every molecule, there's the FDA, and that's their job. But in this new paradigm where you could imagine thousands or tens of thousands of drugs across different modalities, different mutations, different diseases, we need to be able, I think, to have a regulator that is more of a proportionate regulator. I'm borrowing these terms from June Rain, who's the head of the MHRA, the regulator in the UK, who I work with. And I think that, she's really sort of vowed to move more towards an arm's length regulator where there's still regulation, still safety reassurance, but a system that might look more like surgery or stem cell transplants, something where you have an accredited center or accredited system with guardrails and rules, but where the clinicians are able to have a little bit more leeway and look at a child and have a little bit of discretion on, is that child going to die in 6 months or a year or in 5 years, and then really adjust that. Because as a mother, I see all this incredible technology out there, and I know that the risk of not treating Mila, and by the way, the millions like her, is death. Death at 6 years old, 8 years old, 10 years old. It's kind of like, who cares? It's 100% death. And I'm always very curious of why we haven't adopted sort of the risk benefit analysis, or the risk tolerance, maybe is better, from pediatric cancer, where I have friends who've lost their children from cancer. And that conversation is drastically different than the ones that those of us with children with fatal genetic diseases have. So I'd like to say that Mila had end stage genetic disease. When I say that, it changes the conversation completely. We've already established what the risk tolerance is for a dying child with cancer. It's extremely highly experimental, often very risky treatments, because you know what's going to happen to that kid if you don't treat them. Why is that not being applied to children with fatal, 100% fatal and life-threatening diseases? I think starting there and then really thinking about that Millicent fell in many ways, in my mind, between traditional drug development and sort of this highly individualized surgery or transplant, sort of stem cell transplant model. It fell in the middle. So how do we get there? And I think that means regulation first and foremost, with reimbursement at the end, because if it can't be reimbursed, it's not accessible. And we need companies to join this space with the academics, because it's this massive effort. I lived it. And parents selling cookies and lemonade to raise millions of dollars, working with an academic who was never supposed to be taking on the burden of 100% burden for something like this isn't going to work. So both regulation and reimbursement to me are critical.
Lillian, where would you start in your system if you want to make that world happen faster? Where would you start?
I definitely echo Julia when it comes to regulatory. That is something that we have an amazing regulatory department. They didn't pay me to say that, but we have an amazing team at the hospital that each time we go back to them and ask, can we have an exception? Can we have a compassionate use? They jump in and they help. And still at the end of the day, we get a lot of pushback. If we were to be able to have those conversations and set rules moving forward, we would be able to treat vast amount of patients without having to worry about, we're going to receive a denial, we're going to have a pushback. Another issue also that we face a lot in our hospital, because of our patient population, about 60% of them are on some form of government assistance. They're not private insured. Right. So a lot of these tests are not covered by their insurances. We have been very blessed to have private donors, that we have funds that we can afford to have these tests covered for them. But the financial burden is significant, especially on these patients that have been on this diagnostic odyssey for so long. If we were to be able to reduce that financial burden on them, we would also be able to see milestones of improvement, I believe.
And many people may not know that about half the pregnancies in the United States are on federal assistance and not covered by private insurance at all. Where would you start, Mira?
Making diagnostics available to everyone. I think that, the step before the therapy is actually making the correct diagnosis. And there's an access and equity issue there. You know, who are the kids that actually get to hospitals like children's and genetic centers? But also, we have to go through prior approval for every genetic test that we order. And then they're expensive, and, you know, people have deductibles, and it's thousands of dollars. And we argue about clinical utility all the time. And one of the things that any geneticist will tell you is that a diagnosis is useful. You know, these people go, you know, these parents and these families go on these diagnostic odysseys. And if you're even thinking about lowering the cost of healthcare, you know, you have a child with multi-system disease that comes to a hospital like Children's, they may be seen by 7 different specialists who are all going to order a battery of tests. And I have to wonder, like, what's the value of an exome coming in that can help direct your care? So doing something about making diagnostics available is the first thing. The second is education. We have over 1 million licensed physicians in the United States. Half of those physicians are primary care doctors. And I heard earlier today, somebody say that, you know, people will go to their primary care doctor with a result. And the question is, what is the primary care doctor going to do with that result? So we do have to make education available to people, and also variant analysis. You know, a lot of lab tests come back with likely pathogenic or variant of unknown significance. And before you can develop a treatment, you have to be really sure of the condition that you're diagnosing. So I think that there are a few steps along the way. But I think, you know, making a diagnosis, education, and variant curation, I would start right there.
There's been a lot of discussion, certainly in the adult world, about the right to try, particularly in sort of seemingly hopeless situations. I think that it's equally important to think about it in when your parents are representing you, and it's the child that has the right to try, or the parents to decide whether the child has the right to try. I think it's also a key part of the development of all this technology. Well, I want to open it up for comments or questions from the audience, please. So please step up to the microphone, maybe identify yourself.
Tom Burster, I'm a pediatrician geneticist at Maine Health, Maine Medical Center. And I'm co-chair of the policy committee of the International Consortium on Newborn Sequencing. And it's nice to see several colleagues in the office, in the room here, but I want to read some thoughts and encourage the PMC to make several agendas to next year's conference. But President-elect Trump has not won the election because of personality or policies, but rather because the American people were fed up in part with the lack of governmental action to improve their lives. What the American people have soon to learn is that his policies, such as tax cuts to the wealthy and corporations and tariffs, will most likely make their lives worse. And in the introductory letter to PMC Impact Report for this 2023 and 2024, a quote from Dickens, "The Tale of Two Cities. It is the best of times and it is the worst of times. And yes, the best of times is genomic medicine, but the worst of times is our failing healthcare system." And again, I'm a member of this policy subcommittee of the International Consortium, and it is possible to sequence the whole genome of a newborn from the filter paper blood spot at birth, the whole genome. And over 900 conditions, actual conditions, can be identified in that newborn, as well as conditions present later in childhood and adulthood. And the challenge to implementing genome newborn screening into clinical care in the United States, not so much UK, is not the technology, but rather the dysfunctional and fragmented healthcare system in this country. And it's possible to perform rapid whole genome sequencing so that you can have a turnaround within several days, the whole genome, for critically ill newborns. Well, actually brought this up, actually, and identifying maybe 30% of conditions that are genetic. but nobody pays for it. The cost at Rady Children's in San Diego is $7,000. And it's not covered by Medicaid or private payers. But now 16 state legislatures have now mandated supplemental payments for this testing. And I'll just say, to quote from Ernest Hemingway's novel, The Sun Also Rises, "How did you get bankrupt?" And Bill asked, in 2 ways, Mike said, gradually and then suddenly. And we don't want to let our healthcare system go the same way. And so I would ask the leadership for next year's conference to bring Robert Green, who's a professor at Harvard, and he's one of the founders of the International Consortium on Newborn Sequencing, just across the street. And Mira brought up already Wendy Chung, who's the chair at Boston Children's Pediatrics. She's the PI of the Guardian study, and they've already sequenced over 4,000 healthy newborns, and they've identified some remarkable data. So those 2 I would recommend. The other is to bring thought leaders on healthcare reform. Bring people who know how to move forward. Listening to the audience, I've been here for 5 years, and I'm hoping that next year, bringing these topics in, that the auditorium will be filled again. So
Move closer to the microphone, would you? Or maybe it's not turned on. Yeah, thank you for that statement. I'm happy to give up my microphone if you like. Here. It's this one. Here you go.
Oh, okay.
I will not be as eloquent, but I will probably be a little more brief in the sense that I do have a question for Julia. And I think, Paul, you had mentioned a couple of times clinical trials. And given the logistical challenges of an N of 1 or N of few, not being able to have randomized clinical controlled trials, Julia, you've met with tons of regulators. You've had lots of conversations with many of the thought leaders that are in this room and will be here tomorrow. Can you talk a little bit about the challenges of research versus treatment and how research is the solution that's being pursued today, but that's not scalable? Treatment is the scalable part.
Yes, thank you, Kent, for asking. It's a really important question. I know it's a controversial one, but right now, these individualized medicines fall on the shoulders of myself, parents, and on academic clinicians like Tim Yu. And it's an enormous burden in terms of the money, the time, the responsibility, the risk, everything falls on us. And the good news is that in the last few years, since starting the N of 1 Collaborative, myself, Tim, and others, there are 750 members around the world who have raised their hands to say, we want to do what Tim did for Mila, which is really exciting. You know, we didn't know if that void needed to be filled to have an independent hub, and it was, and it's growing rapidly. The problem is that we're not really totally solving the problem because this cannot be scalable. How many parents out there, and I'm being honest, can do, I almost killed myself in a year. I don't know how I raised millions of dollars. I was fortunate to have a wonderful family, wonderful friends, and wonderful people stood up and really felt like they connected with Mila's story. But we know how often this can happen because there's been like 12 kids that have been treated in the last 6-7 years since Mila was treated. that's a dozen, and there's tens of millions of kids that are dying by the age of 5. So we're not even coming close to even scratching the surface. Part of that is because this is considered research and this is considered compassionate use. And what I get told is like, oh, that's such a sweet story that, Tim helped Mila. But the reality is that there's tens of millions of kids dying, and we need a mainstream way of treating disease, not a one-off compassionate use research that's not reimbursable, by the way. That just cannot scale. It's a very simple thing. It's not feasible at scale. And so what does that mean? In my mind, it means that when I look at Mila, she was treated. It was a treat. As a mom, I'm like, she received a treatment. It helped her. It stopped her disease for a year. It wasn't in time for her because she was treated too late, but it was a treatment. And so we need to have companies need to come into this space for no other reason that they need to team up with academics. This needs to be sort of a collaborative model, which means there has to be reimbursement at the end so that we can do this at scale. And so I think we need to shift sort of from this research academic parent alone to a collaborative model. So I don't know, Paul, if you have any other thoughts on that. Or...
So thank you very much for participating. It's been an honor to help you have this discussion. And I hope we've made the argument that personalized medicine and pediatrics go together well. Thank you.
So next we have a panel on a topic, visions for AI and the future of personalized medicine. This is one that PMC has been giving a lot of thought to and is still doing. Our moderator here is Joe Franklin. He's special counsel at Covington and Burling, rather. He comes to that role by way of Verily, and while at Verily, he was a co-chairman of PMC's Artificial Intelligence Working Group. So thank you, Joe, and take it away.
Thank you very much. Thank you very much, Chris. I think if you've been to a conference recently in the healthcare context, I'm sure you've heard an AI panel, and we're very conscious of that here up on stage. We have fantastic array of different perspectives on AI, and I think we're going to try to get into a few specific questions that this context allows us to ask and answer, hopefully. One is, what does AI mean through the very important lens of personalized medicine? And we're obviously at a forum that's uniquely suited to thinking about that question. And then secondly, what should PMC and other stakeholders be doing as we think about advancing the use of AI in healthcare? And what does that mean from a policy perspective? What does that mean from a regulatory and science perspective? So with that, I'm going to let our panelists introduce themselves very briefly, starting with John to my left.
Hi, I'm John Ellithorpe. I'm the president of DNA Nexus. I've been with the company 8 years or so. I run all the product and technology parts of the company. DNA Nexus is a company that focuses on providing a omics platform, a worldwide omics platform that allows people to access, manage, analyze, and collaborate on omics-based data. We started with genomics, but there's been moving from DNA to RNA, to proteomics and such, and incorporating other types of real-world data and clinical data as well. And my background is I have a PhD from a certain institute with a dome across the river, but in astrophysics and cosmology. So I'm not a biologist. I spent most of my time in technology. So I focus on the technology aspects and looking at how we can really advance the field.
Thanks, Jonathan.
Oh, sorry.
I think we have another.
I see it now. Thank you. Good evening, everyone. Jonathan Kish. I lead research sciences at Flatiron Health. Flatiron Health is a real-world data and evidence company. It started about 10 years ago, as probably many of you may know, in building an oncology-specific EMR data system. Over the 10 years, we've used that data to develop real-world evidence, real-world data sets to unlock the power of that real-world data to explore novel treatment options for patients with cancer. The mission of Flatiron is to learn from the experience of every patient with cancer to improve and extend their lives. And I think that is consistent, right, with what we've talked a lot about here today. So look forward to discussing more.
Thanks, Angel.
Hi, my name is Angel Alberic. I am the founder and CEO of Kibim. My background is I'm a PhD in biomedical engineering and computer science. And basically at Kibim, we work on applying AI to transform imaging data, like standard of care imaging, mainly CT, MRI, PET images, into actionable insights. And for doing that, we develop basically algorithms that unlock the information hidden in the medical images, and basically to improve patient outcomes and also maximize drug programs. So we sell to providers. We have already some AI-based FDA-cleared products, mainly in prostate cancer and brain disorders. And then we sell to biopharma in a collaborative framework. Thank you.
Kate?
Hi, everyone. Kate Sasser. I'm the Chief Scientific Officer at Tempus, and I've been in that position for about 2 years. And prior to that, I spent the 20 plus years on the drug development side at companies like J&J and biotechs like Genmab. And I heard Tempus mentioned earlier this morning as potentially an information company. We think of ourselves as an AI-enabled diagnostics company. We'll talk more about what that means, the AI enablement piece in particular. But we're really utilizing things like genomic sequencing, large data sets, integration with EMR, and real-world data to be able to contextualize diagnostics so that physicians can make better decisions for patients, and so that drug developers can make better drugs. So with that, I'll hand it over.
Thanks a lot. Good afternoon, everybody. My name is Okan Akinci. I'm the head of digital technology and CMIO at Roche Information Solutions. Probably many of you know Roche Genentech as a pharma company, Roche as a diagnostics company. We're kind of in between and try to support both our businesses, but also as a standalone company, support clinicians and patients with clinical decision support solutions and remote patient monitoring solutions. Our endeavor started with the realization that our core product portfolio, both on the diagnostic side, but also on the pharma side, with more and more personalized treatment possibilities, requires to support the clinicians and patients with sophisticated IT solutions, today called digital health, to make decisions, basically advancing not only the science as we do it in pharma with upstream kind of drug development integration of AI and IT, but also downstream the utilization of the data for the purposes of making the next best action decision. So as such, I'm looking forward to the conversation. Back to you.
Thanks very much, Okan. So I think to talk about AI, one of the biggest barriers, I think, in having a conversation about AI that's meaningful is to drill into specific use cases. So I'm going to pick on a couple of our panelists to tell us about their favorite potential capability for AI that they hope to see in the next few years. Kate, do you want to get us started?
Yeah, I'm happy to start because I think there are a lot of great, really specific examples that are moving far beyond hype and really becoming reality. So I'll give two quick ones that are, exciting for Tempus and the field at large. One is actually using ECG to be able to diagnose and uncover patients who are at risk for downstream diseases like atrial fibrillation, pulmonary hypertension, and a whole host of other abnormalities. And I think, really the AI comes in play at being able to utilize really large data sets, recognize patterns that we know physicians wouldn't necessarily pick up, and being able to flag that with an algorithm that runs on top of that data set. So that's one example that we like, and that it's made it through the regulatory process and has solved some of those issues. A second one that is coming along and I think very exciting, and Helen and others can speak to, is the digital pathology approaches that are happening today. And I don't know if there's any pathologists in the audience. I work with a lot of pathologists and love them, but I don't know a single pathologist that could view an entire slide and be able to designate whether or not that tumor actually had a genomic alteration, like an EGFR alteration, an ALK, ROS, or others. And so now we see that you can actually, with some of these AI algorithms, you can diagnose some of those alterations from a single slide, meaning you don't have to necessarily do the downstream sequencing. And of course, we need to see this play out in much larger data sets, but this is very exciting.
So is the commonality between those two use cases you mentioned, the EKG and the pathology use cases, the ability to detect signals that humans can't? Is that what AI is adding?
Yes, in both of those, yes. And also, I would say to be able to do things that humans perhaps could do if they had thousands and thousands of hours and thousands of humans, but it's just impossible to be able to scale to the level that computers can.
Great. So, Jonathan, what about you in the research context in particular? What excites you?
Yeah, so the last 10 years at Flatiron and kind of in the real world data ecosystem as a whole, it's still been kind of a bit of a Flintstones age in a lot of ways, right? A lot of the work was done by hand. And what I mean is that much of the rich depth and important clinical features of patients are not contained within structured data elements. They're in the narratives from the physicians and how they're caring for the patients. They're in their smoking status. They're in their performance status. They're in all these places across that health record. And it took an army of individuals historically, right, to read through those charts and extract out that information. Fast forward now to the last several years, and I think there's been a renaissance. We've moved away from simple optical character recognition type of approaches to machine learning algorithms trained on these abstracted data sets, which can now expand the scale with which we can extract that data. And so that's been extremely promising, overall. I think that, is a small example of that, that's been a great starting point. And now we have techniques like generative LLMs, right? And AI technology, and that can accelerate further our ability to extract that information at scale with the same level of depth. So that's the exciting stuff.
Great. That alludes to a topic that we're going to tackle here in a bit, which is generative AI. I think one of the very important things to differentiate. I know, Angel, any other use cases for us to think about as we get started here?
Yeah, I'm really excited on options that can apply AI to sometimes humans have problems when they have to aggregate like multiple parameters. Like for example, when you have MRI, one of the devices we have really created, and when I say device, it's a software, right? But in the end, all prostate MRIs are multi-parametric. That means radiologists have to go through very different images when they read an MRI scan. So they need to aggregate in their brain all the differential, brightnesses, intensities of the images. And that is very challenging for them to learn and to perform adequately on that. So basically, we trained an AI model using biopsies that learn to identify patterns in those data, like are thousands of images in an MRI, that can link the MRI subtle patterns with the biopsy of having a clinically significant cancer. I mean, that means Gleason 7 or above. We are not talking about Gleason 6 here because that is not considered clinically significant. So right now, we already have a tool that's going beyond radiologist performance in identifying those patterns in an MRI. And it's not putting the tool to compete with them, but when you add that kind of algorithm to a radiology reading, if the current sensitivity is 79%, we can grow that up to 90%. And if their specificity is around 68%, we can even grow that specificity. So it's very important when we add AI to a human reading, and it's not decreasing the specificity while increasing the sensitivity. And I think that's showing that's more information that the eye can see right now in the images, and AI is crucial for that.
Angel, can I pick on, or can I push you to describe the different data sources that went into the model you're describing? It sounds like there are MRI and outcomes data as well. What does that look like?
Yeah, so if you want, in my opinion, if you want to really add value to what exists nowadays, I mean, you can decide to train AI models with radiology experts, but if you do that, I mean, then your model will not, be adding value to them in the end. I'm really looking to go beyond their capabilities so that it brings value to them. And the way we did it is basically you take all the MRI data as an input, and then we use like around thousands of patients where we had the MRI, And then we had the biopsy, systematic biopsy, and targeted biopsy. And this is very important because we have many cases where radiologists are not seeing any lesion, but finally it's biopsy positive. And then you need to train the model to identify those cases that are negative for the human eye. And then we can enhance that model. Right now, what I'm describing is basically an already closed algorithm that right now is undergoing clearance. We filed in September. We are pursuing clearance for CADI, CADEX, basically helping for screening. But our goal long-term is being able not to inform only on the cancer detection, but being able to inform on the risk of biochemical recurrence, on the risk of metastasis development in patients. And then we made a partnership in Europe with J&J to be able to early detect those patients at risk.
Okay, and so to achieve those next levels, you'll need to rely on data beyond just comparison to existing or to a radiologist read. Am I right?
Yeah, you need outcomes, definitely. We need to follow those patients in 10 years. The good thing of imaging is that we can go back retrospectively to train the models. We consider them as some digital samples that are immortal, right? So we don't need to destroy the sample. And radiology is already digital by default. So that means we can go back to those PAC systems and rescue many cases.
Thanks. So I think we've talked about a few different use cases in the diagnostic context and in the context of making better use or scaling up our ability to capture data for research purposes. And I think in these conversations, we talk about both the AI tool and the data that's used to develop the tool. John, are we thinking about that in the right way? What's more important, the AI tool or the training data?
From my perspective, I think that let's step back and maybe I'll use a different term instead of the word AI. AI means data-driven programming. And so you're really using data to describe an environment and therefore train the software system to be able to understand what the data represents, what it sees, and therefore, if you can model it, turning the data into knowledge, then you can then use that model to predict things. And so what's really important is the data itself, because if you don't have the right data and you won't expose the right use cases and your system won't fundamentally understand certain things. And so from my perspective, AI technologies are probably going to get commoditized relatively quickly. It's going to be relatively straightforward to, have these different types of transformer models and stuff. You hear a lot of these sort of techniques come out. And I think that in large a number of those will just drive into open source and be freely available, but they're not particularly useful unless you have the right amount of data. And I think that it's not just having the amount of data, but the quality of data is going to be incredibly important. If you look at what happened with ChatGPT, I think that it was trained with billions and billions of dollars and lots of the world's internet, but what really honed ChatGPT was human intervention in the last step of labeling small sets of data to really fine tune those and get high quality data. I think in our space, that's going to be incredibly important to make sure that you have really high quality data to identify and really have precision on predicting the types of things with confidence.
Oken, do you want to make any comments about data and how you're thinking about data and the development of new AI capabilities?
Sure, thank you. We had that question, of course, a couple of years ago when we started considering using AI, more in the old term, I would say right now sense, which is really applying it to data by applying deep learning and try to identify what does the data tell us, exactly to your point, right? I think what right now happens is, of course, we're building all of the short innovation cycle results now into the whole conversation, but our emphasis has been and right now is still exploiting the value that deep learning brings to find connectivity between endpoints and data that you have already collected so that you can roll it out today in clinical practice. And I'll give you just one simple example. So to really illustrate it, CBC, a blood count that everybody gets at least once a year when you do your regular checkup. If you just add age and sex to those few parameters, you can actually predict whether your likelihood of low gastrointestinal pathology is high or not. What does that mean? Well, if you do coloscopy in those who have a high score, the likelihood that these people have indeed a pathology is 10-fold higher. This is not sophisticated science, This is just a pure, very simple approach to combine endpoints with data and try to see what does it tell you. And a lot of critics are there, of course, to say, oh, how are we going to do this in practice, right? So a lot of complicated data are required. Oftentimes when you do algorithms, our approach is right now to say, okay, let's go to the first step, start with the lab data that we have in our customer base, for instance, and add just data from your electronic health record and expand from there. But the point of developing further will, to your question, now require, how do we integrate more data? How do we make sure that the data is really of high quality? And more importantly, how do we get to large scale data that is going beyond just one or two healthcare systems? And there, I think our conversation can also go towards including the LLMs. How do we get to a research environment where more data is being made available so that we don't have 10 different algorithms by 10 small healthcare systems, but perhaps one or two algorithms that are much more sophisticated, meaning higher in diagnostic accuracy, and more importantly, also address the diversity that we need in the patient population.
So I think, one of the questions that comes up as we think about using AI tools, whether it's in the clinical setting or in drug development or medical product development more generally, or other contexts, is how do we understand the performance or the safety and effectiveness or however you're evaluating the model. Can I have any volunteers to address current issues in evaluating models and how, in particular, how we're thinking about demonstrating, for instance, maybe Jonathan, maybe you want to take this and think about demonstrating the validity of a particular type of AI approach for regulatory submissions.
Yeah, so I think the hard part right now is that the regulatory body is just starting to think about this, right? And so the partnership that Flatiron has to collaborate with the FDA to understand what is a threshold of performance of these models if the data will be used in a regulatory framework is an important conversation that we've started to have. So it can be complex technologically, right? Today we heard a lot of talk about sensitivity, specificity. There are other things in there, precision, recall, many different aspects of how you build a machine learning algorithm that are not the simplest things to understand. And if you build one algorithm to identify one data point, and that has a certain recall and precision, what happens when you add that to another data point and another data point? And then how do you look at an ML generated data set in this comprehensive fashion and say what is good enough for a regulatory purpose to use the data? That's the challenge.
So am I right that in some cases it may be for some types of use cases like a diagnostic, you may be able to evaluate it against a gold standard or some existing, an existing reference measurement type and understand how well it's performing. But in other cases that might be possible. Is it what's the right balance? I know Kate, do you want to address this? What's the right balance and under what circumstances, for instance, when we're trying to use an AI tool to provide information in a regulatory submission, is it necessary potentially for the regulator to have access to the training data? And when is that less necessary?
Yeah, I think to be a little bit provocative and push, I think we heard earlier today, like, do we need new frameworks? And I think the answer that you're getting us to is absolutely yes. And we can talk about, do we need, does the regulatory agency need access to training data? Yes, of course. And if we follow the frameworks that are in today, we would do a lot of very stepwise approaches, including, clinical validation and pretty rigid study designs for prospective, randomized studies and follow those, which we should still do. But I think the challenge is very quickly coming where we are going to have a tsunami of these things entering into the LDT space and into clinical practice, and then have to be faced with, which of those actually need to go through the regulatory process, setting aside all the changes we talked about with the election. Which of those need to go through the regulatory process, and how does the agency actually do that at the scale that it needs to? And so that's actually where I think real world data can start to play a huge role of just being able to look at outcomes in almost real time. And in fact, there was an article, I think in JAMA about a month ago, where Robert Califf was actually pointing to this and saying, the entire industry and ecosphere, we heard that word earlier today, the ecosphere is going to have to take some of this on and be able to think about really new ways of looking at outcomes in the real world and showing value of these different therapeutics combined with diagnostics, sometimes combined with multiple diagnostics, and sometimes longitudinal diagnostics that will change over time as the patient is going through a certain journey. So I think we're going to have to come up with completely new frameworks, and it will be perhaps the Wild West for a little while while we're doing that.
Yeah, and perhaps here it's important to acknowledge that I think for many AI implementations in devices, including in diagnostics, there's a pretty well-established pathway for clearance or approval or de novo classification. And FDA in particular has described, I think at this point, reviewing over 1,000 AI-based devices. But I think that what you're alluding to, Kate, is questions that are coming up in the context of kind of novel uses of AI for evidence generation or for other types of for predictive toxicology. Any other comments about those types of use cases? Kate or Oken?
I'm not sure whether that was really the point, because I heard the topic that we have as well, which is like, how are we going to, if we scale the approach to generate algorithms, how are we going to actually get them into clinical routine? My 2 cents to this would be, let's look at how it's done today. And you said rightfully, yes, there are processes in place, but if I go as a diagnostic company, it's a different one than Angel goes as an imaging, let's say, centric algorithm company versus, let's say, DP or others. So everybody has their own angle to it. One thing is we need to treat all of this as data. So we're doing data analytics and create a product that has similarities across the board. So having an understanding what that means is the first step. And the second thing is, our customers, our users, hospitals today are also equipping themselves right now with advanced analytics capabilities. They all have the desire to create novel insights and act on that basis. Right now, they do it with operational topics, but slowly and surely they start getting the taste of, oh, what if we just bring in the clinical data? What if we do this with our EMR data? And suddenly you are already in the same realm. I think what the community needs, and I'm saying here the ecosphere, thank you, the ecosphere, we need to understand what is our common approach here. And it's not just industry going and getting some approvals. It's about how do we want to get AI-based insights into clinical routine? What's the safest way? And it's not going to be a transactional thing. You just get one approval and it's done. It's more a longitudinal establishment of a system that allows you also to monitor these systems. That's what we hear more and more, even from the advanced hospitals who say, yeah, I would like to create our own insights. But if you bring in an algorithm, you need to make sure that drift management is done, bias is already minimized, etc. So there's a totally different type of relationship that's coming also towards us. Because at the end, if I need to manage bias, that means I need to somehow deal with the data, which we normally try to stay away from.
A lot of deep issues here that I think are very, the specific questions that come up for AI depend heavily on the context and on the regulatory status and on the risk use case and so forth. I think we'll maybe focus in on questions that are coming from the audience here in a few minutes, but I want to hit a couple of topics first. One of them is generative AI. And I think that I really want to focus on generative AI for a couple minutes because it is, in my mind, generative AI that is the reason why we're here talking about AI. It's the unpredictability or the, I think, inability for at least many of us to predict that there would be such kind of remarkable capabilities coming out through LLMs over the last few years. And that has focused attention on potential risks and the challenges of grappling with all the different use cases for LLMs, especially potentially in medicine. I don't know, what should we be thinking about in LLMs? And I think, John, I might pick on you to start with, because I think you mentioned in some conversations that we had some more mind expanding potentials for LLMs compared to what I think many of us are familiar with.
So for me, there's like two sort of directions of that where I can imagine AI really being driven forward, right? That changes changes how people work. So one is, I'm in the omics world, DNA and things like that. And LLMs right now have been trained on language, right? And so English language and stuff, we're moving into mathematical languages and things like that to be able to represent. But we have the language of biology, right? We have genomic sequences, we have protein sequences, we have other, if we can figure out how to have sort of these foundational models in that space, that would be very interesting as significant advances towards driving things in silico and being able to do much more in silico. Because once you're in silico, you're orders of magnitude faster in being able to research and development and such. And so that's a fascinating area where, I think we heard earlier today with David and Amgen saying Decode's looking at a foundation model. I think CZI is looking for. So there's going to be a race for these foundation models in biology, especially in the molecular space. So I think that that's going to be a fascinating and interesting world to be able to drive. So that's one part. Another part, since I'm more of a technologist, I'll just say, like, I got my first computer when I was like 12 years old. I've been programming for decades. And I've gone through so many different languages, but fundamentally what I was doing was instead of training the computer to talk to me, I was training myself to talk to a computer. That's what the entire world of computer science and software has been. I think we are now at a space where we can think about having the computer be much more of a, I like the term co-pilot, a co-pilot next to people who do analysis. And so I'm imagining that the world of people doing R programming and MATLAB programming and all these different things moves to a different model where you actually have higher level scientists actually interacting and being able to use this sort of Gen AI sort of technology where you can ask higher level questions and have the system be able to communicate better what it thinks you're trying to do. Now, it's a little bit futuristic, but for now, I think you can sort of see the steps moving that way. And I think those are going to be fundamentally very interesting things if you can take a different way to interact with an analysis environment and then pair it up with foundation models in the biological space. You can imagine just orders of magnitude improvements in how fast we can do science.
Yeah, let's dig into a couple of these capabilities because I'm having a hard time grappling with the possibilities. I think I'm familiar, and I think we're all familiar with ChatGPT at this point. I was impressed by ChatGPT, or it came to my awareness because somebody sent me a ChatGPT written poem about real world data, which I thought, which was like in the style of Shakespeare or something like that, which was amazing 2 years ago, I think, as we all remember. I think I can imagine, and obviously imagine an LLM drafting software code based on some sort of instructions. But what does it look like for an LLM to be trained on genomic data? And what can you do with that capability?
I would say that let's just take a look at what AlphaFold did, right? And so AlphaFold just took sequences and tries to figure out like what the protein structures look like. And I think that those are the types of things where you can sort of fundamentally look and just say, okay, well, if I have certain, like if I can look at certain sequences, let's say I can have DNA sequences, can I start predicting what the RNA could be? And can I start predicting, if you can sort of start embedding sort of epigenetic information and such, like how is a certain DNA sequence going to operate in different types of tumor tissue or tissues and stuff? You can start asking those questions because you can start representing more of that in the system. I think those are the things that you can start doing where you have a low level model that understands certain level predictions like that. But then you have a higher level mechanism where you can ask questions and you don't have to represent the question in exactly some sort of R code or Python code or something like that. You can ask the question and it can interact back with you saying like, okay, are you asking this? Are you asking this? And so can I tell you about this type of thing or this type of thing? And so I think those are the parts that seem very interesting to me, where you can start introducing these bit by bit, but you'll start getting significant advances through that approach.
Thanks, John. I mean, I think that this is really fascinating to watch. And for those of you who aren't aware, I think a must-read document right now is FDA's briefing document for an upcoming meeting of the Digital Health Advisory Committee that goes into regulatory complexities associated with generative AI. And it is just one of the most fascinating regulatory documents, and there are a lot of those in my mind that you can find. Switching to one more topic before we open it up to what I hope to be some audience questions. Kate, do you want to mention any challenges from an operational perspective? And I think that this is particularly relevant in the context of generative AI where one of the fundamental questions is how do we grapple with the unpredictability of the output for an AI tool? And how should we think about operational considerations like the role of the human when it comes to putting these tools in healthcare?
Yeah, I'm happy to talk about a little bit of the last mile, maybe of, once we train a model, or we work with AI to come up with something new, we maybe get it through the regulatory process. How do we actually operationalize it? And I think there's a few things that we could dive into there. I mean, we still know that implementation and deployment is incredibly difficult. And it feels like, should be the simplest part, and yet it's very challenging. So some of the examples we talked about, digital pathology, coming back to that as a really interesting use case for AI. We know that it's incredibly challenging to get that implemented into the workflows with pathologists. And there's a huge discrepancy between some of the places that are represented here, like MD Anderson and Boston Children's and all of the AMCs that have amazing specialists, versus the community centers that may not have access to certain scanners. They may not be able to digitize. So we still have this challenge around just implementation and deployment. I think the other challenge that we see, and at Tempest we deal with this a lot on the physician and healthcare side, is being able to also help physicians with the same entourage and tsunami that we're talking about. And we know in particular that in the community space, physicians, if we stick with oncology as an example, the good news is we're making tremendous progress. There's numerous new therapies emerging. Diagnostics are playing a much bigger role. And yet the bad news is that many physicians are simply overwhelmed with the amount of information that is coming at them every day. It's impossible to keep up. And so I think this is actually a place where AI can also be useful. So we can think about, being able to apply agents into the system. And we're starting to do that, where physicians can ask a simple question like, summarize this report for me. Tell me what I need to do next. Tell me what the guidelines say. Help me connect to a clinical trial. Is this patient applicable for a clinical trial? And today, these agents are already able to do this to a certain level. So you imagine at the pace that we're moving, in another year or 2 years, how much they will improve. So I think it's both a challenge, but also an opportunity.
So I would really encourage anyone who has specific questions for the panelists to step up to the microphone. Can we just make sure the microphone is on? We can hear you. I can reiterate the question for the audience.
They just want the battery, maybe? We can't hear them. Jeez.
And the payers aren't reimbursing this.
I couldn't have phrased the question better. I think this is one of the super important topics for us to get to. So thanks for raising that. Okan, do you want to start us off talking about, I think, what the challenges are from a reimbursement, or just more broadly from a business model for decision support and other types of tools?
Yeah, thank you. And our route, and the question I could have asked as well, but how do we address it today? So today we go to classical route. So if we have evidence that we can influence an endpoint significantly, we can go to long-term route of trying to get reimbursement for it. It's one algorithm at a time. It will take multiple years to get there. We don't think it's scalable. We don't think it will do justice to the innovation that's happening out there. So we are also trying to make sure that, and we have here from market access, my colleague here sitting as well, how do we get the community to the understanding that we have a new category of diagnostic tests? We can call them that way, right? It's a new piece of information based on which we act and make a new decision. And how do we get the value captured for that is a big question that we need to solve as a community. one part that we do instead of waiting until the reimbursement is there to really understand the health economic impact of that piece of information, right? And the example that I've given with the colonoscopy, for instance, is if you're a closed system or kind of an HMO or IDN that is a payvider, right? You have a vested interest making sure that every colonoscopy actually creates value for you. So you can turn that into health economic benefit by introducing that piece. So in that closed system, it works. But in the larger system, I agree with you, if you're in a transaction and fee-for-service environment, adding that piece of information, somebody needs to pay for it. And that will work out currently only if we have models for disease pathways, at least, right? Where parties can come together, as we do it with population health management in hospitals today, to understand what's the impact. Can we create an incremental impact on cost, revenue, or any kind of outcome, but with an economic impact, then we can implement. So I don't have the silver bullet, but I'm just illustrating what the current approaches are, integrating into existing models and trying to go one step at a time, but we need a fundamental change.
It does seem like we have a fundamental challenge about how new information tools are valued in the healthcare process. Any other comments on the panel?
I'll have a comment because I've heard this a lot from a lot of organizations. A lot of organizations have data, and a lot of organizations, and it's likely that a lot of organizations have data of value. But I think what's a challenge is that just having the data and like I haven't. It takes a lot to be able to really understand where you can actually really apply that data to provide value. And that typically requires much more than just the idea of I have data. If you need an entire commercial construct to be able to say that, okay, what am I going to do? What's like, where am I going to take the data? Why am I like, how am I going to target that? Who am I going to sell it to? Another, or monetize it, subsidize it, and such like that. And so I think that like these are things that shouldn't be taken lightly, and the organizations who actually really want to generate like significant value for their data really need to expend significant effort to really understand like where is the value of my data and why do I have, and does it need to be linked with other types of data to really unlock the value or such, because otherwise you're leaving that to whoever you're trying to sell your data to. And I think that I think that the people who are trying to get high quality data sets, a lot of times pharma and things like that, they're getting inundated with requests right now. And to really get through to them, they really need to understand is like, okay, well, like here's a real data set that has value and I've done the work to really identify the value for you. So I think it's really having to go through that effort to really drive and to understand that that data is going to be a commercial business. It's not just a side thing that you might do.
Thanks. Next question.
Is this on? I've thought and written a lot about AI since Dr. Alan Tien in 2015, who was a professor, adjunct professor of applied informatics at Johns Hopkins, where his students didn't like that course because it is difficult. So I've always thought of AI and AI. And I think, and you are the force putting the applied, the machine learning, the AI, artificial intelligence into, and the doctors and medicine is the force to apply the informatics, which you give them in order to assess and diagnose, and then order the treatments and the drugs. And so if you, that's how I look at it. And I think you are amazing. And I think that if you write AI as interventions, it's part of the whole algorithm. And you can spit out at the end of the testing machine, the machine's idea of what the interventions could be, and then medicine can still choose, and the patient can still choose which ones they want to undergo.
Thank you. Thank you for the comment. And the application of AI deserves so much more time than we have today. Any other thoughts?
Just, yeah, two quick thoughts there as the representative of the manufacturer of an EMR system that interfaces directly with the providers. Our goal is not to eliminate the physician from that decision-making process. So two quick examples of things that we're doing are taking all of the NCCN guideline recommended treatments based off of molecular profiles of patients and the complexity that exists there, and showing to the physician, here are the recommended guidelines, just so you're aware, so you're not missing anything, right? You make the final call on how you want to treat that patient. But that program is important, right? Because it's such a complex and it's ever changing, right? These guidelines update constantly. So offering that to them, I think, is a stepping stone. At the same point, I think we can all attest, and we showed in our data day in and day out, that there are gaps and disparities in NGS testing, right? There's a lot of patients that should be getting it that aren't getting it. Can we use the tools of AI, right, to search through those notes, search through that structured data again, and give a nudge to that provider to say, look, based off of these characteristics of the patient, it might be appropriate to test this patient, not to sponsor any specific testing group, right? But those are the types of things that we're implementing now at the point of care to be able to improve and close those gaps.
Yeah.
Thanks. Next question.
So you have briefly discussed the challenges of the deployment process of AI tools. And in this context, some researchers have suggested the interest of, in the context of healthcare, using the approach of having specific information labels, like the ones that are used for prescription medications, where you can inform about the process of the capability of the particular AI tool, which is also very important, I think, for the explainability of these kind of tools in the context of healthcare. So I would like to know your opinion about this potential complementary information in the process of deployment. Thank you.
Yeah, so there's a question around labels for transparency and explainability for models, a feature of some regulatory frameworks like ONC's framework. Anyone want to address explainability or transparency information? Is it feasible? I guess one question that comes up in my mind is it feasible to provide this information in readily digestible form in the way that has been proposed with kind of nutrition style labels, or is that going to be overwhelmed by the massive scale of AI tools as they become more common?
Yeah, I think it's a good idea and it's a good approach. I think there's a wide range already in the ways we're using AI and what's behind the actual algorithms in terms of how much information you can provide. And because we were just talking about the challenge of deployment and information overload, yes, we can provide more information labels on how the model was trained, what data sets were used, the explainability of what's driving the algorithms. But I think that will also add to the information overload. And so, and we are also pretty rapidly getting to a point where I think some of the AI algorithms you will be able to reverse engineer them, but you're talking about so many parameters that it becomes just another matter of scale. So it's not so much that it's completely black box and we have no idea all the different features and parameters that are going into the algorithm, but the fact that it's thousands and thousands and thousands in the future very quickly. And so just the scale that it would take to be able to evaluate each of those is going to rapidly move beyond anyone's capability. So it's explainable, but not necessarily verifiable in the ways that we're used to when we're dealing with much simpler parameters.
Okay.
A brief addition, perhaps. I think, yes, labeling will become important. How detailed is the question, right? So we see, for instance, a trend right now also in lab testing. You could simulate lab test results, right? If you have enough data and you can actually match it with endpoints, like specific ranges of lab results, you could actually measure out of vital parameters today, the BNP range that a patient has. So for assessing heart failure, for instance. But if you present that result, you need to make sure to label it as this is an AI assessed lab result because it's never been measured. It has been calculated based on an AI algorithm that was trained on data before. So I think what we can definitely expect is that anything that has touched AI should probably get a label like this has been touched by AI, just to make sure that we have the skepticism as a clinician as well to go back and say which aspect, right? What was it exactly that I got here? Because we need to be vigilant that we don't run also into a second or third AI winter, if you want, in healthcare. As somebody said today, we're still in the hype curve here. And with Gen AI is the same thing. Interesting enough, that's the most advanced stage, if you want, of AI, but the acceptance among our customers is much higher for LLM-based tools right now than it is for classical deep learning. Why? Because they see exactly what they get, right? In deep learning, you don't see it. It's a black box. What happened? You can trust them more than you can trust LLM today. There's also this people aspect, I think, that we need to cover. How do we understand and change the mindset towards building trust? It's not what you just see. It's also about how do we understand how AI has evolved and which part is actually more robust and which part is probably more to be confounded with skepticism.
So flagging content that is created with AI,
which has touched, yes,
is kind of a base level
exactly
need in your mind.
Yeah, sometimes I refer to algorithms as the new drugs, ideally, because we have a level of I would say rigorosity when we are developing drugs, where what are the clinical studies going on, how many patients were included in this study, different details that you might find. We even have clinical trials at Gov that's giving a lot of information, but we don't have access to the details of how we train the AI models. And I'm a manufacturer. I would be happy to open my technical descriptions because that's for the benefit of all of us, right? I mean, the same information I'm sharing with the FDA. I mean, this is my data set for training. This is the different ethnicities, the ages, whatever. This would help me really compare performance, because right now, what we manufacturers can specify in the 510K summaries, it's very limited. I mean, sometimes it's very limited and you really don't know the performance results, what's going on there, how was trained, what was the data set, what were the characteristics. And once you deploy that in clinical practice, the doctors need to know, whether a given patient is in specifications or is out of specs. And I think we need to have kind of open technical descriptions of AI so doctors can know whether it's on the responsibility to use it in a certain group of patients, or is the manufacturer the one that's accountable on that scenario?
Yeah, I wonder because for equitability purposes, as well as there are going to be gaps in the data that people train on, and we have to live with that. And so I wonder if we're going to enter a world which looks a little bit like the drug business and stratifying patients with biomarkers. Should we have data stratifications for applications of AI-based predictive algorithms because they work better and they've been identified better because they've been trained on certain sets of data tied to certain patient populations? And so we may end up in that world where there'll be some level of regulatory confidence levels in certain populations, and then you'd have to publish that, and then those AI algorithms would be somewhat prescribed based on whether you're in that population or not, and then you'd know that if you're outside that population to be lower confidence and such.
So I think the commonality is information that allows the user to sort through just a massive number of AI tools, understand how to use them. I think we have time for one more audience question.
Hi, my name is Brad Power. I'm the founder of the Cancer Patient Lab and of the Cancer Hacker Accelerator for startups that are doing cancer diagnostics and navigation. When I look at AI, I see two things. We're at the Personalized Medicine Coalition. Personalization means N of 1. N of 1 means possibly going outside those NCCN guidelines you were referring to, because I've gotten functional testing that tells me that something that I have is unique and is an off-label treatment. That's just one aspect, but I just would be interested in hearing someone talk about something no one has talked about here, which is the use of Gen AI to empower patients and caregivers, consumers of healthcare, to be full co-pilots with their medical team.
Sorry, to be full co-pilots with what?
Their medical team.
With their medical team, thank you. So yeah, a great question. Any comments on deploying AI, deploying generative AI in this case, in this context?
Yeah, I can offer a few tidbits that I think are interesting. So one is your point about getting to an N of one type of approach for oncology or for other diseases. I think we're rapidly seeing the onkification of diseases like cardio and other places. So N of one is I think coming. We just heard in the pediatric symposium right before, the panel before, about how challenging that is from a commercial perspective and from a drug development perspective. I do think that we are getting to a point from a data and Gen AI perspective where it is becoming more of a reality, meaning the things that were ultra rare or unable to be kind of identified earlier on are now at our fingertips. And we see patients being able to leverage these tools, even the general LLMs like OpenAI, ChatGPT, Perplexity, Claude, pick your model that you want to use. We already know that patients are actually uploading data that they have, lab results, genetic test images, and that these models are able to actually give them, in some cases, some pretty good advice that they weren't necessarily able to have through the typical medical system. So I also think that we're going to rapidly move to a space where patients, maybe not all patients, there's quite a large disparity among different patient groups, but patients are already seeking out these applications. And so I think companies will also have to think about patients as their own data controller and being able to make available to patients a lot of these data models and tools that we're able to.
I think, yeah, leverage for pharma and others. Okay, so I think that brings us almost to the end of the session. I want to just thank my panelists for a fantastic conversation. We couldn't possibly address every single topic here. Thank you to the audience for questions, and I hope everybody had a great day.
On behalf of TNC, I would also like to thank the panel. And also, given that it's late in the day and everyone here has stuck it out, I would like to buy you a drink at the Museum of Fine Arts. There'll be a shuttle to take us to the museum. If you haven't been there, it's a treat all by itself. And again, thank you.
Yeah.
That's what I